Hello there and welcome back to the sleepy science. You're lying there, half buried in blankets, your phone casting that gentle blue glow across the ceiling. Outside, the world is doing whatever it does, buzzing neon in cities, creaking crickets in suburbs, or just waiting for sunrise in whatever time zone you've drifted into tonight. Inside you, though, something very small is very, very busy. In fact, there are tens of trillions of little compartments inside you right now, each one humming with activity. Within those compartments, cells, to be a bit more precise, there lives a tiny structure that looks like a squishy bean under a microscope. It has wrinkles like a raisin and its own set of tools, and it's been called the powerhouse of the cell so many times, even your high school biology teacher was probably sick of hearing it. Still, that label doesn't even begin to capture how bizarre, bossy, and essential mitochondria really are. Every breath you take is for them. Every calorie you eat gets funneled through their miniature machinery. Without them, your heart would stop before you could even Google, "What do mitochondria do?" And without exaggeration, you'd die in about 10 seconds if all of them suddenly quit. So, before you get comfortable, take a moment to like the video and subscribe, but only if you genuinely enjoy what I do here. Also, drop a comment with your current location and local time. Now dim the lights, maybe turn on a fan for that soft background hum, and let's ease into tonight's journey together. You're drifting now, but you're also heading inward, shrinking in your mind's eye, spiraling down through layers of skin and sinew, past organs and blood vessels, until you're hovering somewhere inside your own cells. Think soft pink interiors and warm, jelly-like fluid. You land gently on a thick, pill-shaped structure. It pulses slightly, alive but not in a way you can quite understand. This is a mitochondrion, singular. It's part of a vast inner network, a cellular grid full of these power hubs, and they're not exactly team players. Each one is semi-autonomous, meaning it does its own thing while still working for the greater good. A little like a stubborn coworker who happens to be brilliant at their job. They come in thousands per cell, especially in energy-hungry ones like those in your brain, heart, or muscles. Cells that just sit around doing not much, like fat cells, still have them, but far fewer. If you think of your body as a city, mitochondria are the power plants. And just like power plants, they aren't always clean or quiet. When they're churning out energy, they also release tiny reactive byproducts, like a candle releasing a little smoke along with light. Here's the wild part. Mitochondria weren't always part of your cells. That's a story for later. But it's worth knowing up front that they're basically adopted. They moved in a long, long time ago, and now they're indispensable. But for now, zoom closer. You float through the outer membrane, which is smooth and lets certain things come and go, like water or small molecules. Inside, it gets more complicated. You see a second, inner membrane that's folded like a velvet curtain. Those are the cristae. They're the secret to how mitochondria do what they do. Inside those folds, enzymes and proteins run a tight operation. They're part of what's called the electron transport chain, which you'll explore soon enough. But for now, just think of them as tiny factory workers handing off packets of energy like some molecular relay race. This whole process ends with the production of ATP, adenosine triphosphate. Think of it as the rechargeable battery pack of life. Your muscles, neurons, liver, all of it burns ATP to get things done. Without it, even blinking would be an impossible task. And here's something that might make you blink. Your body generates around your own weight in ATP every day. Not all at once, of course. It's made and recycled in a frantic cycle, where each ATP molecule might get used and rebuilt hundreds of times per day. Talk about hustle. Historically, the first major discoveries of mitochondria happened in the 1800s. Scientists squinted through crude microscopes and noticed some strange blobs hanging out inside cells. They didn't know what they were seeing, just that these thread granules moved differently, almost like they had a job to do. Eventually, biochemists figured out their purpose, and by the 1950s, the term powerhouse of the cell was slapped onto textbooks and stuck. But mitochondria are full of quirks, even beyond their productivity. Did you know some of them can move around inside the cell, like they're searching for the best spot to work? Or that they can fuse with each other to share resources, or split apart if one gets damaged? Basically, they're multitaskers with a touch of drama. And even though they're inside your cells, they act like they remember being on their own. You see, mitochondria have their own DNA, different from your nuclear DNA, and we'll talk about that DNA party in just a bit. For now, just let the image settle. Your cells are alive with movement, and nestled in each one, these wrinkled little beings are turning oxygen and nutrients into the very essence of life. But here's a reality check. If something goes wrong with your mitochondria, it's not like having a glitchy app. It's more like having your power grid flicker during surgery. Mitochondrial diseases are some of the most devastating and often untreatable conditions around. They can affect the brain, the muscles, the eyes, even your hearing. And sometimes the symptoms don't appear until decades after birth. So as you lie there, drowsy and soft around the edges, know this: every heartbeat, every thought, every dream you slip into tonight, it's all because your mitochondria showed up for work. And they're not even getting paid. You don't usually think about ancient history while brushing your teeth or scrolling through grocery apps, but your cells remember things that stretch back over 1 billion years. And floating right now inside nearly every one of them is a relic of a very strange evolutionary deal. That mitochondrion you're picturing, wrinkly bean, doing its thing, well, it used to be its own creature. A free-living bacterium, minding its own microbial business, until one fateful moment when it moved in. The story is murky and still fiercely debated. Historians still argue whether the host cell swallowed the bacterium intentionally, or whether it was just a case of single-cell surprise real estate. Some think the larger cell gobbled the smaller one and somehow didn't digest it, like a snake swallowing a rodent, and then deciding to be best friends. Others believe it was more of a merger, two struggling microbes realizing they had something the other needed: shelter and food in exchange for energy production. Whichever way it happened, what came next was revolutionary. The engulfed bacterium, believed to be a close relative of modern alpha protobacteria, started working for its host. Over time, they grew more dependent on each other, like two odd roommates who eventually just start sharing toothbrushes and Netflix accounts. Genes from the bacterium transferred into the host's main DNA stash, tying their fates together permanently. Eventually, it stopped being a separate organism and started being mitochondria. This fusion of two life forms is known as the endosymbiotic theory, and it's one of the most beautiful, slightly creepy ideas in biology. Your cells are, in a way, not entirely your own. They're ecosystems, communities. And this merger is why complex life like plants, animals, and people exists at all. Without mitochondria pulling their weight, our cells wouldn't have the energy to be anything more than blobs. No movement, no multicellular organization, no you. Let's zoom out for a second. Imagine Earth about 1.5 billion years ago. No trees, no fish, no continents like we know them. Just a vast soup of single-celled organisms bobbing around in shallow oceans. And then this merger. One bacterium climbs inside another and doesn't get kicked out. And suddenly the host cell can do things its cousins can't. Like burn fuel more efficiently, divide faster, specialize, evolve. Fringe theorists even go so far as to suggest that mitochondria aren't just helpful, they're in control. That they subtly direct, cellular decisions by releasing signals, initiating cell death, or influencing development. Some researchers call mitochondria the cellular sentinels, not because they're conscious, of course, but because they sit at the intersection of energy, metabolism, and mortality. It's a fun twist. You're the host, but deep inside your body, the descendants of ancient bacteria might be steering the ship just a little. And this arrangement hasn't stayed consistent. Over millennia, mitochondria shrank. Most of their genes, the instructions that told them how to do everything, migrated into the nucleus of the host cell like a tenant slowly packing up and handing over keys. Today, mitochondria still have their own DNA, but it's minimal. They rely on the cell for a lot of help, proteins, enzymes, and even signals telling them when to divide. They're not free anymore. They're domesticated. Still, the relationship is complicated. Mitochondria retain just enough independence to be weird. They grow and divide like bacteria. They respond to stress signals like they're still on their own. And under a microscope, when they fuse or split, they move with a kind of eerie coordination, like a synchronized swimming team made of blobs. Here's a quirky bit for your sleepy brain. Some scientists discovered that mitochondria can form extensive networks inside cells. Instead of floating separately like beans in soup, they can stretch and link into dynamic webs. In neurons especially, these webs can extend down the long arms of nerve cells, almost like the mitochondria are commuting to work. Which makes sense. They're the fuel providers. The more demanding the cell, the more mitochondria show up. In one particularly memorable experiment, researchers used a fluorescent dye to track mitochondria in living cells. They watched, real time, as they zipped along microtubules, dodging obstacles, merging with others, and then peeling away again. It was less like watching organelles and more like spying on some bizarre microbial freeway. Of course, not all cells play nice. Sometimes, mitochondria get hoarded, damaged, or ignored. In certain parasites, they've been pared down to almost nothing, like protozoan squatters living off the host with the smallest possible backpack. And in other life forms, mitochondria have taken wild turns. Some anaerobic organisms, those that don't use oxygen, have bizarre versions of mitochondria that can't make ATP at all. They still need their little roommates, but for different jobs. This is why mitochondria are so fascinating. They're universal, but not uniform. They've been bent and reshaped by 1 billion years of evolution, like a Swiss Army knife that keeps gaining tools you didn't ask for. Some researchers even study mitochondria in giant amoebas and sea cucumbers to see just how flexible they can be. Turns out, very. One more historical nugget before you drift further. Scientists first started seriously connecting mitochondria to evolution in the 1960s, thanks to the tireless work of microbiologist Lynn Margulies. Her theories about endosymbiosis were initially mocked or dismissed. The idea that two separate life forms merged into one seemed absurd at the time. But she kept going, and eventually her ideas helped reshape how we understand life itself. Today, textbooks quietly include what once seemed too radical to publish. So here you are, lounging in the present day, built by an alliance that started before any animal walked the land. Your cells are cities. Your cities are run by ancient bacteria, and the only reason you can breathe, think, or stretch your toes is because two microbes decided not to kill each other. What a strange piece, Triddy. You're still floating inside the cell, drifting gently past the folds of the inner mitochondrial membrane, and now it's time to talk about the real reason this whole structure exists: ATP. Not a particularly sexy acronym, sure. It sounds a little like an airport or a tax form, but adenosine triphosphate is the stuff that keeps you alive, literally. Without it, your body would grind to a halt faster than a phone on 1% battery. So let's start slow. Imagine that everything your body does, from the blink of an eye to a 5-mile jog, from digesting lunch to sending a thought through your brain, costs ATP. It's your body's only accepted form of energy payment. No cash, no credit, just ATP. When you eat food, your cells break it down into smaller parts, mostly glucose and fatty acids. Then, with a little oxygen and a lot of finesse, mitochondria take those parts and turn them into this golden currency. Now, picture a little factory line humming away in the inner membrane of the mitochondrion. This is the electron transport chain, and it's a bit like a Rube Goldberg machine built by chemistry nerds. Electrons, stripped from the food molecules you've broken down, are passed along a series of protein complexes embedded in the membrane. With each handoff, a little energy is released. Not too much, not too little, just enough to pump protons across the membrane and build up a gradient. Historians still argue whether Peter Mitchell, the scientist who proposed this chemiosmotic model in the 1960s, was a genius or just lucky. But eventually, his wild theory, that a proton gradient could power ATP production, was proven true, and he even won a Nobel Prize. You may not remember his name, but every breath you take is living proof that he was onto something. Back inside the mitochondrion, those protons now rush back across the membrane through a special enzyme called ATP synthase. Think of it as a microscopic turbine, spinning rapidly with each surge of protons. And every spin of this molecular machine presses together one add molecule, adenosine diphosphate, and a free phosphate group, snapping them together like puzzle pieces to create ATP. Click, click, click. Every second, billions of these clicks are happening inside you. It's like a biochemical printing press, churning out tickets that let your cells buy whatever they need to stay alive. Want to contract a muscle? Spend some ATP. Need to move a nerve signal? That'll cost you. Just maintaining your internal temperature and keeping your heart beating eats up pounds of ATP per day. Now here's a fun and slightly alarming fact. Your body uses more than its own weight in ATP every 24 hours. But don't panic, it recycles it constantly. Like an energy coin that gets spent, collected, recharged, and spent again. The same ATP molecule might do a dozen laps through your body in a single minute. The mitochondria are so efficient at this that they produce 90% of all the ATP in your body. Sure, cells can do a little bit of energy making without them. There's a process called glycolysis that happens in the cytoplasm, but it's like running your entire house on a solar-powered calculator. It's cute. It's not enough. This is why muscle cells, especially those in your heart, are packed with mitochondria. They don't get rest days. Your heart never clocks out, not even when you sleep. So it needs a steady supply of ATP, which means a massive demand for mitochondria, which means that those cells are absolutely crammed with wrinkly little bean machines. There's even some quirky diversity in how mitochondria work across different cell types. In liver cells, for example, mitochondria double as detox experts, helping to break down ammonia and process alcohol. In brown fat cells, especially in bobbies and hibernating animals, they're geared for heat production, burning fuel inefficiently on purpose to keep the body warm. Kind of like setting a controlled fire in a fireplace instead of turning on the lights. And speaking of inefficiency, there's a weird outlier in ATP production called uncoupling. It's when the mitochondria decide not to produce ATP at all, but to just burn the energy and release heat instead. Your body uses this trick to regulate temperature, and it may even play a role in weight regulation. Some researchers are exploring it as a potential anti-obesity strategy, though, as always, the science is still a bit fuzzy, and there's no magic mitochondria pill just yet. Let's go microscopic again for a moment. You glide past an ATP synthase molecule and it's spinning like a windmill in a hurricane. You imagine yourself the size of a proton, flung across the membrane, zipping through the turbine. Every turn generates another ATP, and for a moment, you almost hear the soft tick of productivity. Click, 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 like rain falling on a roof. But here's the kicker. Even this flawless process can break down. Mitochondrial diseases often involve defects in the electron transport chain, which means electrons leak, ATP production falters, and harmful byproducts build up. The energy drops off a cliff, and the consequences can affect everything from movement to cognition. Some disorders are inherited, others develop over time. Even in perfectly healthy people, ATP production naturally declines with age. Your mitochondria wear down. Their membranes stiffen. Their enzymes get less efficient. It's like the factory doesn't shut down, but some of the machines get squeaky and start to miss a beat. And yet, every moment of your life so far has been powered by these organelles. From your very first heartbeat in the womb to this exact second, mitochondria have been quietly fueling you like devoted, slightly overworked employees. If they could talk, they might ask for a raise, or at least a decent night's sleep. You're drifting even deeper now, and the view gets stranger the closer you get. The mitochondrion is no longer just a bean-shaped blob. It's a whole world of folds, walls, compartments, and enzymes packed in like an overbooked red-eye flight. And the deeper you go, the more intricate it gets. Let's take a proper look at those folds, shall we? They're called cristae, and they're the wrinkled inner lining of the mitochondrial inner membrane. From a design standpoint, they're a masterpiece of space-saving architecture, kind of like stuffing a queen-size comforter into a carry-on. The more surface area you can cram into that tight space, the more room there is for all the important proteins and enzyme complexes that run your energy system. If mitochondria had smooth walls, your cells would be outrageously inefficient. You'd probably burn out trying to blink. Under the microscope, cristae vary wildly. In liver cells, they're more tubular and tightly packed. In heart cells, they're longer and more linear, like carefully stacked lasagna noodles. This isn't random design. It's an energy-specific layout. The more energy a cell needs, the more complex and tightly curled the cristae become. Form follows function, and in this case, the function is keep this human vertical and conscious. What's amazing is that these folds aren't static. Cristae can actually move. They change shape depending on what's going on in the cell. Under high energy demand, they expand. During low activity, they relax. Imagine if your house could grow new walls during a dinner party and shrink back afterward. That's what your mitochondria are doing while you scroll Instagram or attempt yoga. And speaking of quirky real estate, mitochondria are divided into several key regions. You've got the outer membrane, which lets small molecules in and out like a sleepy nightclub bouncer. Then there's the intermembrane space, that tiny zone between the two membranes where proton gradients build up like pressure in a shaken soda can. And at the center is the matrix, which sounds like a sci-fi title, but is really just the innermost chamber where enzymes float around and the Krebs cycle unfolds. Ah yes, the Krebs cycle, that iconic swirl of arrows and chemicals you once saw on a biology poster and immediately tried to forget. It's the part where mitochondria convert fuel from your food, glucose, fats, amino acids, into high-energy electrons that get fed into the electron transport chain. Think of it as the prep kitchen, chopping ingredients and handing them off to the cooking line. Fringe researchers have even suggested that the geometry of cristae might carry information, like some kind of structural code that influences how mitochondria behave. That's still speculative, but there's no denying the folds do more than look pretty. They're functional origami at the atomic level. Now there's a reason this internal complexity matters so much. Without it, you wouldn't have multicellular life. Back in the day, when cells were simpler and energy needs were modest, something like a flat cell membrane was fine. But when organisms started getting more complex, muscles, nerves, brains, they needed more juice. And mitochondria, with their Christie, stepped in like a genius architect who could make a tiny studio apartment into a power plant. Historians still argue whether Christie evolved once or multiple times in different lineages. It's possible that not all mitochondria share a single ancestral membrane style. Some primitive eukaryotes, for example, have mitochondria with different inner structures, flat, tubular, even bizarre loops. It's kind of like the mitochondria across species are trying on different hats to see which works best. And if you want a truly niche trivia tidbit to lull you deeper into sleep, in some rare species, the Christie don't even fold inward. They fold outward. It's like trying to cram your laundry into the drawer by pressing from the inside out. Why? No one knows. Biology loves its oddballs. Back in your own body, though, the Christie are doing what they've done every day of your life, facilitating energy production at a level so microscopic you can barely imagine it. Each crystal hosts dozens of protein complexes, like ATP synthase, cytochrome C oxidase, and NADH dehydrogenase, working in teams. These names are about as exciting as a jar of beige paint, but don't let that fool you. They're among the most efficient machines on Earth. And nestled inside these Christie, there's even more complexity. Some sections act like loading docks for metabolic substrates. Others help signal the rest of the cell when things go wrong. Some even hold onto calcium ions, releasing them in tightly regulated bursts that affect everything from muscle contraction to neurotransmitter release. That's right, your mitochondria are micromanaging your calcium. And they don't work in isolation. Mitochondria often cluster near areas of high energy demand inside the cell. In neurons, they hang out at synapses. In muscle cells, they camp near contractile fibers. They're like mobile chargers stationed at your busiest internal airports. Their dynamic behavior means that even the Christie themselves aren't locked into one layout. They reshape in response to damage, temperature changes, or signals from the nucleus. If a section becomes dysfunctional, the mitochondrion might fuse with another to share Christie. Or it might fission, split into two, to quarantine the bad parts. That's not just fascinating, it's vital. Dysfunctional mitochondria are cellular liabilities, and your cells know it. So yes, your cells are energy factories, but they're also part air traffic control, part crisis management, part urban planning. And those tiny folds you never think about. They're the quiet heroes keeping the lights on in your internal cityscape. Some scientists even speculate that the fine-tuning of Christie structure might help explain age-related decline. As we get older, our mitochondria tend to develop swollen or disorganized Christie. The one-sorterly folds collapse, the membrane potential fades, and ATP production slows. It's like a once bustling shopping mall, now full of broken escalators and flickering lights. But right now, your Christie are doing just fine, folded, functional, and flowing with protons. It's not glamorous work, but it's non-stop, and it's happening inside you night and day as you sit, stand, snack, or snooze. Let's all take a moment to appreciate the humble fold. You're still adrift inside the mitochondrion, hovering just above the matrix, the inner sanctum of this cellular engine room. And now it's time to talk about something that makes mitochondria even weirder than they already are. They carry their own DNA. That's right. Tiny, independent, slightly rebellious strands of genetic material, just doing their own thing, right in the middle of your energy factory. Most of your DNA lives securely in the cell's nucleus, neatly coiled and tightly regulated, like a vault of instructions guarded by layers of security. But mitochondria, nah, They keep a backup copy of their own manual, small, circular, and self-contained. It's like if every coffee shop in town also printed its own tiny cookbook, just in case the internet went down. This mitochondrial DNA, mtDNA, is different. It's short, just 37 genes compared to the 20,000 plus found in the nucleus. But don't let the size fool you. Those 37 genes code for critical components of the electron transport chain and for the RNA machinery needed to make those proteins. Without them, the whole ATP production process would crash like a power grid during a summer blackout. And here's the thing, that DNA doesn't come from some mystical central archive. It's bacterial in style and origin, circular, like the chromosomes found in prokaryotes, and prone to making copies of itself without asking the nucleus for permission. That's another relic of its ancient roots. Remember, mitochondria were once free-living bacteria, and their DNA is one of the last pieces of evidence. Historians still argue whether mitochondria hung on to this DNA out of necessity or just stubbornness. Some say it's because those few genes need to be close to where their protein products are used, in the mitochondrion itself. Others suspect it's evolutionary inertia. These genes simply never got around to moving into the nucleus like the rest. And of course, keeping their own DNA means mitochondria get into trouble the same way any DNA does, with mutations. Unlike nuclear DNA, which has elaborate repair mechanisms, mtDNA is more exposed. It's located right in the middle of where free radicals and reactive oxygen species are flying around, like fireworks near a paper towel warehouse. Mistakes happen. Damage accumulates. And over time, this can lead to cellular malfunctions and chronic diseases, everything from muscle degeneration to neurodegeneration. Here's something fringe but intriguing. In some organisms, mitochondria have way more genes than ours do. There's a weird little plant called Silene canica, whose mitochondrial genome is nearly 400 times larger than the human version. Scientists still don't know why. It's not like the plant runs marathons. Its energy needs are modest. But for some reason, its mitochondria hoard genetic material like dragons on a DNA pile. And here, another twist. Mitochondria don't just carry DNA, they also have their own ribosomes. These are smaller than the ribosomes found in the rest of the cell, and they look a lot like bacterial ones, which, again, is a smoking gun for their bacterial ancestry. They're not just part of the cell, they're descendants of something once entirely separate. Now let's talk about how this DNA behaves. In humans, each mitochondrion can have several copies of its genome, and each cell can have thousands of mitochondria. That means your body holds many thousands of mitochondrial genomes, all coexisting, sometimes harmoniously, sometimes not. There's a term for when things get messy: heteroplasmy. That's when different versions of mitochondrial DNA exist within the same cell, some normal, some mutated. And the balance between those versions can determine whether a cell functions normally or starts to break down. If the proportion of damaged mtDNA crosses a certain threshold, the cell starts to suffer. Energy output drops. Symptoms appear. This is one reason mitochondrial diseases are so variable and so hard to diagnose, because it's not just whether the DNA is damaged, but how much of it is. Scientists are actively studying ways to manipulate this. There's research into mitochondrial replacement therapy, a process where faulty mitochondria in an egg cell are swapped out for healthy ones from a donor. This has led to the media-friendly, ethically loaded term, three-parent baby. The child inherits nuclear DNA from two parents and mitochondrial DNA from a third. Welcome to the future, where you might literally have extra help powering your cells. But don't worry, this isn't about cloning humans or designing super athletes with elite mitochondria. At least, not yet. For now, the goal is to prevent certain devastating diseases, though naturally, bioethicists are already lining up with questions like, how far is too far? Who gets access? And what happens when something goes wrong? And while we're here, there's something else you should know. Mitochondrial DNA is inherited exclusively from your mother. That's right. Your mitochondrial lineage is maternal, going all the way back, unbroken, through your mom, your grandmother, your great-grandmother, and so on, deep into prehistory. Sperm do have mitochondria, but they're located in the tail. And when the sperm fuses with the egg, the tail gets left outside. Only the mother's mitochondria make it into the embryo. Which means your cellular energy factories are a direct biological gift from your maternal ancestors. Like heirloom jewelry, but with fewer diamonds and more proton pumps. This maternal inheritance has made mitochondrial DNA a powerful tool in evolutionary biology and forensics. It mutates slowly, and because it doesn't mix like nuclear DNA, it provides a clean genetic trail. Scientists use mtDNA to trace human migrations, identify ancient remains, and even solve cold cases. But it also raises some deeply strange questions. What happens when a mutation enters that maternal line? Does it quietly pass down through generations before finally causing symptoms in a child, grandchild, or great-grandchild? And could we ever edit mitochondrial DNA directly to stop that legacy? Gene editing tools like CRISPR are still clumsy when it comes to mtDNA. Unlike nuclear DNA, mitochondria don't like being poked with molecular scissors. They tend to shut down, like grumpy employees when corporate installs new software. Still, research presses on, therapies are being tested, questions remain. But one thing for sure, you are powered in part by ancient foreign DNA that decided to stick around and help. And somehow this bizarre, bacterial legacy still keeps your lights on. You're probably not thinking about your mom's mitochondria when you're deciding what to binge-watch next or when to reheat those leftovers. But deep down, humming away in every one of your cells, her legacy is churning out energy molecule by molecule, day in and day out. That's right. When it comes to mitochondria, you didn't get a 50-50 deal. You got a maternal monopoly. Every single mitochondrion in your body comes from your mother. 0 from your father. None. Zip. You could say you're powered by maternal energy, and not just in the poetic "thanks for making me" sense. It's literally baked into your biology. When sperm and egg meet, the egg doesn't just provide half the genetic material, it also supplies the full suite of cellular organelles. Sperm arrive like overenthusiastic delivery guys, speedy, specialized, and very focused on the job. But they don't bring much with them besides DNA. Their mitochondria are tucked into the tail, which gets left behind after fertilization. It's like showing up to a potluck with nothing but a fork. Historians still argue whether the evolutionary reason behind this mitochondrial matrilineality is about quality control or conflict avoidance. One theory suggests that if you mixed mitochondria from both parents, you'd risk cellular warfare, different mitochondrial populations competing inside the same cell. Not exactly a recipe for harmony. So, evolution may have decided to keep things simple. One mitochondrial lineage per person, passed through the mother's egg like a family recipe too fragile to copy twice. This maternal-only inheritance opens up all kinds of fascinating possibilities, and a few unsettling ones. For one, it means your mitochondrial genome is likely almost identical to your mother, your grandmother, and so on, with only minor mutations adding variety over generations. That makes mtDNA an excellent tool for tracing ancestry. It's how scientists figured out, for instance, that all living humans share a common mitochondrial ancestor who lived roughly 150,000 to 200,000 years ago. More on her later, but yes, we're talking about the so-called mitochondrial Eve. But before we climb into ancient evolutionary trees, let's come back to something closer to home. What happens when mitochondrial inheritance goes wrong? Because when it doesn't always happen evenly. This is where the idea of the mitochondrial bottleneck comes in, a process that's still a bit of a black box, but here's the general idea. When a mother passes her mitochondria on to her children, not all of her mtDNA copies make the trip. Only a select few end up in each egg cell. It's as if her mitochondria get put through a filter, and only a small, random handful gets chosen to move forward. This bottleneck can amplify mutations. If a mutation is present in a small portion of her mitochondria, it might get weeded out, or it might suddenly become the dominant version in the next generation. The result is unpredictability. One sibling might inherit a high proportion of faulty mitochondria and develop disease. Another might be completely unaffected. Same parents, same genetics, different mitochondrial lottery. And it's not just rare diseases either. There's growing evidence that mtDNA variants influence common conditions like diabetes, neurodegeneration, and even mood disorders. Some researchers are exploring whether maternal mtDNA could be part of why certain illnesses run in the family in odd, uneven ways. It's not always about the genes in the nucleus. Sometimes, it's the tiny powerhouses carrying baggage from the past. There's also a fascinating gendered twist. Even though men carry and use mitochondria just like women do, they don't pass them on. So natural selection acts primarily through the female line when it comes to mitochondrial traits. This creates a bit of an evolutionary blind spot. Traits that harm males but don't affect females as much might not get weeded out efficiently. This concept has been jokingly referred to as the mother's curse hypothesis. Here's a quirky example. In some insects and mammals, researchers have found mitochondrial mutations that reduce male fertility but leave females totally unaffected. Since only females pass on mtDNA, those mutations can persist in the population without much resistance from natural selection. Your mitochondria might be fantastic energy producers, but terrible wingmen. On the human side, there's ongoing debate about whether this phenomenon affects us significantly. Some studies have hinted at links between certain mtDNA mutations and reduced sperm motility, but the evidence isn't conclusive. Still, it's one of those strange little evolutionary quirks that make you wonder just how many secret rules biology is playing by. And speaking of rules, the maternal inheritance of mitochondria also comes with ethical and scientific implications. Remember mitochondrial replacement therapy, the process where faulty maternal mitochondria in an egg are swapped out for healthy donor ones before fertilization. It's already been used in rare cases to help women with mitochondrial diseases have healthy children. But it also breaks the classic mold of reproduction. The resulting child has nuclear DNA from both parents and mitochondrial DNA from a third person, the egg donor. Cue debates. Are these children three-parent bobbies? Technically, yes, though the mitochondrial DNA makes up less than 0.1% of their total genome. But still, it's a new frontier in inheritance, one that's forcing society to reimagine how we define biological lineage. Some governments have approved this technique. Others have banned it outright. The science is solid, but the public's comfort level still catching up. Meanwhile, back in your cells, the mitochondrial matriarchy continues. Every heartbeat, every breath, every flicker of thought is powered by machinery handed down from your mother's side. You didn't get to choose it. You can't upgrade it, but it's yours. A little maternal legacy packed into every cell, as faithful and fragile as a hand-me-down locket. So the next time you feel your heart racing, or your legs powering up a flight of stairs, know this. It's not just you. It's a lineage. A line of mitochondria stretching back through your mother's body and her mother, before that, all the way to a woman who lived long before history began. And she too had folds in her mitochondria. She too made ATP in the dark. Let's shift the lighting for a moment. Not the cozy bedside lamp you're under, but the interior glow of your cells. The warm thrum of mitochondria isn't just about energy. These tiny organelles are exquisitely sensitive to your state of being. They respond to your stress. They react to your sleep schedule or lack thereof. And they most certainly notice when you inhale a frappuccino and half a cinnamon roll before noon. In other words, mitochondria are not cold little robots mindlessly pumping out ATP. They're dynamic, emotional, even if we're being metaphorical, which we absolutely are. They adjust themselves constantly based on what's happening both inside and outside your body. This responsiveness makes them essential, yes, but also vulnerable. Because when your life goes out of balance, they're often the first to feel the hit. Start with stress, that ever-present companion of the modern world. Whether it's a looming deadline, an argument, or just trying to parallel park while someone watches, your mitochondria feel it. When your brain senses stress, it sends out cortisol and adrenaline, those fight-or-flight messengers. These chemicals don't just make your heart race, they tweak how your mitochondria function. They push the cell into survival mode, altering the way energy is distributed. For short bursts, this is helpful. You want your muscles ready to leap away from metaphorical tigers. But chronic stress, that's a different beast. Long-term exposure to stress hormones can reduce mitochondrial efficiency, impair ATP production, and increase the production of reactive oxygen species, the infamous ROS. Think of ROS like exhaust fumes from a car. They're normal, expected, and manageable in small amounts. But too many of them build up, damaging mitochondrial membranes, disrupting the electron transport chain, and mutating DNA. It's like trying to power your house with a generator that's slowly catching fire. And yes, your cells do have antioxidant defenses, but they weren't designed for the kind of stress modern humans endure 24/7, from news alerts to late-night doom scrolling. And then there's sleep, sweet, elusive sleep. When you're well-rested, your mitochondria hum along smoothly, energy production is balanced, repair processes run on time. But when sleep is erratic or insufficient, especially deep sleep, the restorative kind, your mitochondria struggle. Studies show that sleep deprivation reduces mitochondrial respiration and increases ROS. Even one bad night can tilt the balance. Historians still argue whether sleep evolved primarily for brain recovery or cellular maintenance, but either way, mitochondria play a starring role. During sleep, cells ramp up repair mechanisms. Damaged proteins get cleared. Mitochondria perform quality checks. If you shortchange your sleep, you short-circuit this process. The damage builds up, and over time, that means fatigue, fog, and worse. And we haven't even touched nutrition yet. What you eat isn't just fuel for your stomach. It's instruction for your mitochondria. Every morsel of food gets broken down into molecules they can burn. But not all fuel is created equal. Diets high in processed sugar, trans fats, or artificial additives can overload mitochondria, especially in cells that regulate metabolism like those in your liver, pancreas, and brain. A spike in blood sugar, for example, forces mitochondria to work overtime, producing more ATP than necessary and releasing more ROS as a byproduct. It's like asking your car to redline every time you drive to the corner store. Over time, this leads to insulin resistance, inflammation, and cellular exhaustion. Meanwhile, a balanced diet rich in whole foods, Healthy fats and micronutrients tends to stabilize mitochondrial output, smoother, slower, more sustainable. There's even evidence that certain nutrients, like coenzyme Q10, magnesium, and B vitamins, support mitochondrial function directly. These aren't miracle cures, of course, but they serve as essential cofactors in the energy production process. Without them, the whole biochemical orchestra goes a little out of tune. And let's not forget physical activity, that other lifestyle cornerstone. Mild to moderate exercise boosts mitochondrial biogenesis, meaning your cells literally grow more mitochondria in response to increased energy demands. Regular movement teaches your body to make and maintain better powerhouses. But too much intense exercise without recovery, you guessed it, ROS overload, mitochondrial burnout, and systemic fatigue. This is why that no pain, no gain mindset can backfire, especially if you're underslept, overstressed, and underfed. It's not about pushing your body until something snaps. It's about respecting the delicate balance your mitochondria maintain every single day. And speaking of burnout, let's talk about cellular fatigue. This isn't just feeling tired after work. This is when your mitochondria can't keep up with your demands. It's what underlies chronic fatigue syndrome, fibromyalgia, and even long COVID in many cases. Emerging research suggests that in these conditions, mitochondria either can't produce enough ATP or become hypersensitive to stress signals, shutting down at the first sign of trouble. Fringe researchers have even proposed that mitochondrial dysfunction might underlie more of our common ailments than we realize, from mood swings and migraines to anxiety and ADHD. The link isn't always direct, but it's persuasive. When your energy systems falter, everything else feels harder. Thoughts slow, muscles ache, emotions fray. You're not just tired, you're energetically bankrupt. And then, of course, there's the feedback loop. Mitochondria affect your mental state, and your mental state affects your mitochondria. Stress disrupts energy production, which makes you feel worse, which increases stress, which damages mitochondria further. It's like trying to climb out of a hole while someone keeps shoveling dirt on your head. But the good news, your mitochondria are remarkably responsive to change. Start getting better sleep, managing stress, eating real food, moving more, and they start to bounce back. The cells sense the shift. The environment improves, and the mitochondria adjust. They're not quick to give up. They're ancient, adaptive, and weirdly loyal. So tonight, as you wind down and exhale a little deeper, know this. You're not just relaxing your mind. You're helping a billion tiny engines ease off the throttle. You're telling your mitochondria, you can rest now too. And they're probably relieved. The lab is cold, always cold. Bright overhead lights hum with clinical indifference as you peer through the microscope, adjusting the fine focus knob with slow, deliberate movements. Your breath fogs slightly against the eyepiece. And there they are, those same mitochondria, dancing and flickering across the stained backdrop of the cell, revealing just enough of themselves to tease the story underneath. You're not imagining this. They actually do flicker. Little pulses of light as calcium surges in and out, or as membrane potentials shift. The cell is alive, and its energy factories are visibly glowing. Mitochondria under the microscope are surprisingly beautiful. They're long, snaky, constantly in motion. Sometimes they form networks, sometimes they isolate, curling into themselves like they're hiding from something. This isn't a chaotic mess, it's choreography. Controlled, responsive, and in some cells, almost elegant. In fact, when scientists first observed mitochondria moving, they were baffled. In the late 1800s, German pathologist Richard Altman saw these bioblasts under primitive staining techniques and recognized they weren't static. They pulsed and shifted inside the cytoplasm, unlike any organelle previously observed. At first, people thought they might be parasites, little invaders living off the host cell. And honestly, given their bacterial origin story, that wasn't such a bad guess. But over time, better microscopes and better dyes told a clearer story. Mitochondria aren't just blobs. They're active, responsive, and oddly social. With advanced techniques like fluorescence microscopy and live cell imaging, we can now watch mitochondria drift along cellular highways, stretch like taffy, merge into long tubes, then split again like cosmic noodles. Their shape is constantly changing. It's called mitochondrial dynamics, and it's one of the hottest subfields in cell biology. Because how mitochondria move often reflects how healthy, or how doomed, a cell is. Fringe fact to tuck under your pillow: mitochondria move along structures called microtubules, which are basically the cell's internal railway tracks. They use motor proteins, tiny molecular engines with adorable names like chymsin and dynein, to walk along these tracks, carrying mitochondria to wherever energy is needed most. A bit like Uber drivers, but with far more punctuality and a lot less attitude. And the destinations vary. In neurons, mitochondria travel all the way down long axons to fuel synapses, the places where electrical signals jump between cells. In immune cells, they gather near membranes when a threat is detected, helping to amplify the cell's inflammatory response. They're not drifting aimlessly, they're part of a strategy. Historians still argue whether mitochondrial mobility evolved from the bacterial flagella system, or whether it's a separate innovation born inside eukaryotes. But what's clear now is that cells use this mobility to maintain efficiency, directing mitochondria to where ATP is needed most, or to where damage control is urgent. And that brings us to something darker: damaged mitochondria. Because just like humans, mitochondria have bad days. They get leaky, lose their membrane potential, or start producing too many reactive oxygen species. When this happens, they send out distress signals, chemical beacons like cardiolipin flips or the release of cytochrome C. These aren't just symptoms. They're flags. The cell takes notice. And sometimes it responds with mitophagy, a form of cellular recycling where broken mitochondria are swallowed up and digested. It's a bit gruesome, but efficient. Better to lose one dysfunctional mitochondrion than to let it bring down the whole system. Watching this happen under a microscope is like witnessing a microscopic soap opera. Healthy mitochondria may fuse with a damaged one in an attempt to share resources, mitochondrial solidarity if you will. But if the damage is too great, the cell chooses cleanup over compassion. The broken bits get tagged with proteins, dragged into lysosomes and digested. It's swift, merciless, and necessary. Some researchers have taken this observational science to artistic extremes. Using advanced super-resolution microscopy, they've captured stunning images of mitochondrial networks glowing in colors: red for membrane potential, green for calcium flux, yellow for metabolic activity. The pictures look like city lights at night, like neural maps, like deep space nebulae. One could argue mitochondria are the universe within your cells, flickering with life, fatting with stress, always in motion. Here's a lighter note to balance the mood. In 2019, a team of scientists managed to film mitochondria engaging in a sort of dance inside human stem cells. They twisted, collided, rebounded, and settled again, like microscopic waltzers at a molecular ball. It was mesmerizing, like watching ink swirl in water, only with far more consequence. And what about structure? Under transmission electron microscopy, mitochondria reveal their famous double membrane in exquisite detail. The outer membrane is smooth, porous. The inner membrane, as you know, is folded into cristae. But at high enough resolution, you can even see the protein complexes studded along the folds, like studs on a leather jacket, tough and incredibly efficient. These complexes are responsible for nearly all your ATP, and scientists have started building 3D models of them to better understand how changes in their shape might lead to disease. Which brings us to the diseases visible under the lens. Mitochondrial myopathies often show up as ragged red fibers in muscle tissue, cells swollen with dysfunctional mitochondria that cluster under the membrane like squatters refusing to leave. In some neurodegenerative diseases, researchers observe fragmented mitochondrial networks, signs of stalled dynamics and cellular decay. Alzheimer's, Parkinson's, Huntington's, each one has a mitochondrial story written somewhere in its pathology. But imaging is not just about diagnosis, it's about hope. With real-time observation, scientists can now test drugs on living cells and watch the mitochondria respond. Do they recover, reconnect, resume ATP production? Some experimental compounds have shown promising results, restoring mitochondrial motion and reducing ROS in models of aging and metabolic disorders. It's still early, but the future glows, just like those mitochondria on the slide. So the next time you hear someone casually call them the powerhouse of the cell, remember this. They're also performers, dancers, couriers, first responders, and under the right lens, tiny stars lighting up the night inside you. You float there, watching the mitochondria glide gently through the cytoplasm like lanterns on a darkened sea, but suddenly something feels off. One of the mitochondria looks swollen, its shape distorted. Its inner membrane is torn. It pulses irregularly. This isn't just an underperforming organelle. This is a cellular emergency. And when mitochondria go rogue, things unravel fast. Welcome to the war zones of the cytoplasm, where mitochondrial dysfunction can trigger chaos that ripples through the entire cell like a thrown stone in a still pond. The mitochondria aren't just producing energy. They're also monitoring threats, managing death signals, and helping cells decide what to do when things start going sideways. And when their internal wiring shorts out, they can bring down the house. At first, the signs are subtle. A drop in ATP, a rise in reactive oxygen species, maybe a sluggish fusion-fission cycle. The mitochondrial network begins to fragment like a tightly knit community falling apart. Cells that once thrived on smooth energy flow start to sputter, unable to meet basic demands. In most tissues, this is bad. In organs like the brain or heart, it's catastrophic. One of the most studied mitochondrial breakdown scenarios is in neurodegenerative diseases. In Parkinson's, for example, damaged mitochondria accumulate in neurons, especially those in the substantia nigra, the region responsible for dopamine production and movement control. Mitochondria there start leaking ROS, and the cell's ability to clean them up through mitophagy breaks down. It's like garbage piling up in a city with no sanitation department. Neurons begin to die, and movement becomes impaired. Historians still argue whether mitochondrial dysfunction causes these diseases or is simply a tragic side effect. Some scientists point to mutations in specific genes, like PINK1 and Parkin, which are involved in mitochondrial quality control. When those genes fail, broken mitochondria hang around too long, and the cell slowly poisons itself. The brain isn't the only battlefield. Mitochondria are involved in heart failure too. Cardiomyocytes, heart muscle cells, have the highest density of mitochondria in the human body. When these mitochondria falter, ATP production drops, calcium regulation skews, and the finely tuned rhythm of your heartbeat stutters. The cell tries to compensate, but eventually the damage is too great, and whole sections of the heart may weaken. Now enter the immune system. White blood cells also rely on mitochondria, not just for energy, but for triggering inflammatory responses. In fact, mitochondria can release mitochondrial DNA into the cytoplasm, which your immune system misidentifies as foreign. Remember, mitochondrial DNA still looks suspiciously bacterial. This can cause the body to sound the alarm, unleashing cytokines and initiating inflammation. In autoimmune diseases, this mechanism might go haywire. Some researchers believe that faulty mitochondria leaking DNA or ROS can act as false flags, leading the immune system to attack healthy tissue. In lupus, for example, mitochondrial fragments are often found in circulating blood, and they seem to aggravate the body's own defenses. Mitochondria in these cases might be unintentionally instigating friendly fire. Let's zoom into something more microscopic but equally troubling: cancer. For decades, the prevailing theory was that cancer cells don't use mitochondria for energy. They rely on glycolysis instead, even when oxygen is available, a phenomenon called the Warburg effect. But this idea is changing. We now know that many cancer cells still use their mitochondria for certain tasks: biosynthesis, ROS signaling, and cell survival. In fact, some tumors rely on mitochondrial metabolism more than others, especially those in oxygen-poor environments. There's even fringe speculation that some cancerous behavior might originate from ancient mitochondrial pathways, primitive survival tricks turned destructive. Some rogue mitochondria may even shift their metabolic profile to help tumors grow faster. Instead of powering healthy cells, they start enabling uncontrolled division and invasion. They switch sides. Of course, not all mitochondrial dysfunction leads to full-blown illness. Sometimes it just results in fatigue, brain fog, or a lingering sense that your body is running at 70% no matter how many hours you sleep. This kind of subclinical mitochondrial burnout is hard to detect, harder to diagnose, and easy to dismiss as just stress. But deep inside, the cellular engines are sputtering. Cells try to fight back. They increase antioxidant production. They shift metabolic pathways. They signal for help. And sometimes, if things are truly dire, the mitochondria initiate a final act: apoptosis. That's right. They have the power to trigger the death of their own cell. It's not sabotage, it's damage control. If a cell is beyond saving, the mitochondria flip the internal switches that begin a safe, clean self-destruction protocol. More on that drama in the next section, but for now, understand this. The mitochondria are not just background machinery. They're guardians, strategists, and when pushed too far, saboteurs. Their decline can be subtle, like a dimming light bulb, or explosive, like a fuse leading to cellular death. And they don't go down alone. One failing mitochondrion can signal others. Dysfunction spreads like rumor through a small town. Entire cell systems collapse. But sometimes, just sometimes, the opposite happens. One healthy mitochondrion can merge with damaged ones, share its contents, and stabilize the system. It's a quiet kind of heroism, playing out trillions of times a day in your own body. So when we say mitochondrial dysfunction is at the root of many chronic diseases, it's not hyperbole. It's a battlefield report. There are places in your body right now where mitochondria are quietly winning, others where they're struggling, and maybe even a few where they've already lost. But you're still here, which means the war, for now, is still being fought, and many of your little powerhouses are still holding the line. You wouldn't expect something as small and wrinkled as a mitochondrion to wield the power of life and death. But here we are. Among all their jobs, power production, metabolism management, calcium buffering, there's one role mitochondria take particularly seriously: the final say in whether a cell lives or dies. That's right. These humble powerhouses are also the executioners, quietly holding the trigger to apoptosis, or programmed cell death. It sounds grim, but apoptosis isn't some dramatic murder scene. It's a clean, quiet shutdown, cells folding up their internal machinery, cutting off communication, and fragmenting themselves into neat little packages to be swept away by the body's cleanup crew. No swelling, no inflammation, no chaos. Just a silent bow out, orchestrated with almost surgical precision. And mitochondria are at the heart of it. When they sense that a cell is beyond repair, too damaged, too mutated, too infected, they take it upon themselves to initiate the shutdown. They do this by releasing certain proteins into the cytosol, most famously cytochrome C inside the mitochondria. Cytochrome C plays a perfectly innocent role in the electron transport chain. But outside the mitochondria, it's a death sentence. Once released, cytochrome C binds with a protein called APAF1, forming a complex known ominously as the apoptosome. This structure then activates a cascade of enzymes called caspases, which go on to dismantle the cell from the inside out. DNA gets chopped. Structural proteins are cleaved. It's a full demolition job, and all of it starts with a whisper from the mitochondria. Historians still argue whether this apoptotic pathway evolved as a defense against viruses, as a method for shaping developing tissues, or both. Either way, it's essential. Without it, your body would be riddled with malfunctioning cells. During fetal development, for example, apoptosis helps sculpt organs, removing the webs between your fingers and toes, shaping the folds of your brain. Without it, we'd all look a little more like jellyfish. But apoptosis isn't just about development, it's also about control. Your body makes billions of new cells every day. Without a way to remove the defective or outdated ones, you'd be drowning in your own biological debris. Mitochondria act as the internal referees, weighing metabolic signals, oxidative stress, and DNA integrity to determine whether a cell should keep going or bow out. Now here, where it gets interesting and slightly terrifying, some cells resist apoptosis. Cancer cells, for instance, often mutate the very pathways that lead to programmed death. They block cytochrome C release, disable caspase activation, or overexpress survival proteins like BCL2, which literally plug up the mitochondrial pores that allow death signals to escape. The result? Cells that should die don't. They keep dividing, hijacking your systems, and building tumors that ignore every natural warning sign. On the flip side, in neurodegenerative diseases like Alzheimer's and ALS, apoptosis may be too eager. Neurons begin to die even when they aren't beyond repair. The mitochondria become hypersensitive, interpreting normal wear and tear as irrecoverable damage. It's the cellular equivalent of pulling the fire alarm over burnt toast. In fact, mitochondria's control over apoptosis has made them a target in cancer therapy. Some experimental drugs are designed to resensitize mitochondria in cancer cells, to open the pores, release the death signals, and force the cells to self-destruct. It's a delicate game. Too much apoptosis and you risk damaging healthy tissue. Too little and the cancer keeps growing. But if you can aim the signal just right, the mitochondria can do what they were always meant to: maintain balance. There's also a related, more chaotic process called necrosis, which used to be thought of as accidental messy cell death. But scientists now recognize there's a hybrid form, necroptosis, that is partially regulated, and mitochondria are involved there too. They help determine which path the cell takes: clean exit or violent rupture. It's like choosing between quietly leaving a dinner party or flipping the table on the way out. Let's not forget their role in immunity either. When a cell is infected with a virus, sometimes apoptosis is triggered not to protect the cell, but to protect the whole body. Infected cells sacrifice themselves to stop viral replication. Mitochondria, once again, are the ones making the call. It's a risky move, but it buys time for the immune system to mobilize. And here's something fringe but fascinating. Researchers have recently found that mitochondria can influence non-apoptotic death pathways too. Some cells can die through ferroptosis, a process driven by iron and oxidative stress. Others go through pyrotosis, which involves inflammation and is especially common in infected cells. In all these cases, mitochondria are deeply involved, either as instigators, victims, or sometimes even spectators. What's clear is that mitochondria don't just manage energy. They manage fate. They're the cell's inner judge, jury, and occasionally executioner. And somehow, they do this without any consciousness, just a finely tuned sensitivity to signals, damage, and time. From an evolutionary perspective, this makes sense. Cells that could self-terminate when infected or damaged likely gave their organisms a survival advantage. Cleaner tissues, less cancer, better control. But it also means that a lot rests on the health and discernment of your mitochondria. If their judgment gets clouded by age, by mutation, by stress, the consequences ripple outward. And yet apoptosis isn't always the end. Cells around the dying one often benefit. The fragments are cleaned up by phagocytes, resources are recycled, space is cleared. In a strange, quiet way, cell death fuels life. Your skin renews itself this way, your gut lining, your immune cells. Apoptosis is one of the few things that keeps your internal systems from turning into a chaotic cellular hoarder's den. So tonight, as you lie there under your blanket, know that somewhere inside you, thousands of cells are deciding whether to stay or go. Not with drama, not with fear, just quiet evaluation. A few will choose to end gracefully in service of the whole, and the mitochondria, ever watchful, will carry out the sentence. Because sometimes, keeping the lights on means knowing exactly when to turn one off. Let's imagine for a moment that your mitochondria are employees. Not corporate types in stiff suits, but wiry, practical engineers running around your cells, fixing leaks, tightening valves, spinning turbines. Most of the time, they work around the clock, quietly converting fuel into ATP with mechanical grace. But what if you wanted them to be better, faster, more efficient, less prone to burnout? Well, in a strange and promising twist, you can actually train your mitochondria. Not with a whistle or a clipboard, unfortunately, but through certain stressors and behaviors that push them to grow, divide, and evolve. Just like lifting weights builds stronger muscle, exposing your mitochondria to the right kinds of pressure can boost their performance. Not overnight, but steadily, consistently. And exercise is where it all starts. Physical movement, especially aerobic exercise like walking, jogging, swimming, signals to your cells that more energy is needed. More energy means more ATP, which means your mitochondria have to step it up. In response, your body starts a process called mitochondrial biogenesis, the creation of new mitochondria from scratch. The conductor of this process is a protein called PGC1-alpha, pronounced PGC1-alpha, if you want to impress someone at a biology-themed dinner party. When activated, this protein tells your cell nucleus to start transcribing genes that will assemble fresh mitochondria. More mitochondria means more capacity, more resilience, and better energy output across the board. Historians still argue whether early humans evolved large numbers of mitochondria in response to long-distance running or climate adaptation. But either way, the connection between movement and mitochondrial performance is ancient. Our ancestors didn't run marathons for medals. They ran for food, for shelter, for survival. Their cells adapted, and so do yours. But the benefits of exercise don't stop with quantity. It also improves mitochondrial efficiency, how well each mitochondrion processes fuel and oxygen. Studies show that trained muscles use oxygen more effectively, burn fat more cleanly, and produce less oxidative stress than sedentary ones. It's like upgrading your old coffee maker to a sleek new espresso machine. Same ingredients, much better results. Now, if exercise is a steady kind of training, Then intermittent fasting is like high-intensity interval training for your cells. Going without food for periods of time creates mild stress that forces your body to shift metabolic gears. Instead of running purely on glucose, it starts burning fat for fuel, releasing ketone bodies, which mitochondria love. Ketones are cleaner burning than glucose, generating less oxidative damage and more ATP per molecule. They're like premium fuel at the cellular gas station. Fasting also triggers autophagy, the process by which cells clear out old, damaged mitochondria, making room for healthier ones. It's spring cleaning at a microscopic level. Some fringe enthusiasts even combine fasting with cold exposure, plunging into ice baths or frigid lakes to further challenge their mitochondria. Cold triggers the activation of brown fat, a type of fat tissue rich in mitochondria that burns energy to generate heat. The shock of cold forces your mitochondria into overdrive, not to build muscle or endurance, but to keep you warm. This adaptive thermogenesis is an ancient survival mechanism, and though it's still being studied, early evidence suggests it can boost metabolic flexibility and increase mitochondrial density. Let's not forget the opposite extreme, heat exposure. Saunas, hot yoga, and even warm baths raise core body temperature, which has its own set of mitochondrial effects. Heat stress activates heat shock proteins, which help stabilize and repair damaged mitochondria. These proteins are like cellular therapists, calming and supporting mitochondria under pressure. Still, it's not just about physical extremes. Mitochondria also respond to psychological cues. Practices like mindful breathing, meditation, and yoga can reduce cortisol, improve mitochondrial membrane potential, and even influence gene expression related to energy metabolism. Think of it as mental cross-training for your cells. When you breathe slower, your mitochondria aren't just watching Netflix in there. They're adjusting, restoring, recalibrating. And here's something quirky: hormesis. It's the idea that small doses of stress make you stronger. Exercise, fasting, heat, cold, all of these are hormetic stressors. They challenge your cells just enough to provoke growth without tipping them into damage. It's a balancing act, one that your mitochondria know well. Too little challenge and they stagnate. Too much and they unravel. But just enough, they thrive. That's why mitochondrial training isn't about punishing your body. It's about strategic nudges, encouraging adaptation without pushing into burnout. It's why long walks might serve you better than daily HIIT workouts. Why skipping breakfast once in a while might reboot your energy systems more effectively than any supplement. Speaking of supplements, yes, some people try to hack their mitochondria with coenzyme Q10, alpha-lipoic acid, NAD+ precursors, and the like. There's some science behind these, especially in people with mitochondrial disorders or aging-related decline. But the truth is, none of them hold a candle to what your own behaviors can do. No pill builds more mitochondria than a brisk 30-minute walk. No powder outperforms a regular sleep schedule. What really matters is consistency. Mitochondria don't respond to fads. They don't care about one heroic workout or one perfect day of eating. They're like quiet coworkers who notice patterns. They get stronger when you treat them well over time, when you move often, sleep deeply, eat clean, and challenge them just enough to keep them sharp. So tonight, as you lie still, wrapped in comfort and silence, your mitochondria are still adjusting. They remember the stairs you climbed earlier. They noted the salad you almost didn't eat but did anyway. They're cataloging, responding, adapting, becoming more efficient in the dark. And if tomorrow you take a cold shower or skip that snack or go for a jog, they'll notice that too. Not with words or feelings, but with chemistry, enzymes turning, membranes shifting, energy flowing more cleanly through the invisible engine rooms of your body. They're listening, training, getting better. All they need is a little push and a little patience. It starts as a slow drag. You wake up already tired, your brain wrapped in gauze, every thought moving through molasses. You sit down to work and realize you've read the same sentence 5 times. By midday, your limbs feel like they're moving through syrup. Sound familiar? Welcome to the fog, the peculiar mental fatigue that doesn't quite count as sickness but absolutely doesn't feel like health. And here's the quiet culprit that might be behind it all: your mitochondria. When people think of fatigue, they often picture weak muscles or low blood sugar, but increasingly, researchers are pointing fingers at mitochondrial dysfunction. Not the dramatic, disease-level collapse we've explored earlier, but a more subtle, creeping kind of slowdown. A dip in ATP production here, a touch of oxidative stress there, and suddenly your energy just goes missing. This isn't just physical exhaustion, it's brain fog, and yes, your mitochondria are involved. The brain, for all its quiet mystery, is an energy hog. Though it makes up only about 2% of your body weight, it consumes about 20% of your resting metabolic energy. That's a lot of ATP, and that means a lot of pressure on your brain's mitochondria to keep the lights on. In healthy brains, mitochondria are stacked up like firewood in neurons, especially at the synapses where signals are sent and received. They power the movement of neurotransmitters, the restoration of electrical gradients, the recycling of cellular waste. But when mitochondria fall behind, due to stress, inflammation, poor sleep, or chronic illness, your brain starts rationing energy. Things get fuzzy, focus slips, emotions wobble. Historians still argue whether brain fog is its own discrete syndrome or just a symptom cluster wrapped in a name. But mitochondrial dysfunction is one of the few common threads that show up in conditions where brain fog is reported: fibromyalgia, Lyme disease, chronic fatigue syndrome, long COVID, and even depression. The fog may not be in your imagination, it may be in your cells. Let's zoom in. In states of chronic inflammation, immune cells release cytokines, those famous little messengers that tell the body when it's under attack. But cytokines also affect mitochondrial function. They can increase oxidative stress, suppress ATP synthesis, and even damage mitochondrial DNA. In the brain, this creates a vicious cycle. Inflammation weakens mitochondria, which reduces energy, which impairs detoxification, which increases inflammation again. This is why even seemingly minor illnesses can leave you wiped out for days. When your immune system kicks into gear, your mitochondria temporarily shift focus away from energy production and towards stress response. They scale back the power grid to conserve resources. That's fine for a short-term battle. But when this state lingers, like it can after a viral infection, the fog settles in. And it's not just physical inflammation. Emotional stress does a number on your mitochondria too. Prolonged stress increases cortisol, which alters glucose metabolism, and as we saw earlier, leads to the production of more reactive oxygen species. In the brain, this can mean mitochondrial burnout. Some studies have even shown that patients with depression have lower mitochondrial function in key brain regions. That heavy, listless feeling. It might be mitochondrial exhaustion dressed up as emotion. Now here's a quirky twist. Caffeine, your beloved fog-busting sidekick, has a complicated relationship with mitochondria. In moderate amounts, it actually improves mitochondrial efficiency, encouraging more ATP production and less fatigue. But overdo it, and you risk triggering the very stress signals that suppress mitochondrial function. That third cup of coffee might feel like a productivity boost, but your mitochondria might see it as a call to emergency mode. And if you've ever crashed hard in the afternoon, eyes burning, brain stuttering, heart thudding, that's not just a sugar crash or poor sleep. It's a power crash. Your mitochondria have been running non-stop all day, and if they didn't get the nutrients, oxygen, or rest cycles they needed, they wave a tiny flag and start dialing back output. So what can you do when the fog rolls in? Beyond the usual sleep, hydrate, move trifecta, there are some interesting mitochondrial tricks. Movement, even gentle movement like stretching or walking, can stimulate mitochondrial biogenesis and ATP production, especially in the brain. It might feel counterintuitive. You're tired, so why move? But movement signals demand, and demand drives production. Nutrient support helps too. Your brain mitochondria especially love omega-3 fatty acids, B vitamins, magnesium, and antioxidants. These don't work like energy drinks. They're not instant fixes, but they're the raw materials your mitochondria need to keep up. Without them, it's like asking a factory to operate with no spare parts. Some fringe researchers are exploring photobiomodulation, a fancy term for exposing the body, or brain, to near-infrared light. The theory is that mitochondria absorb certain wavelengths of light, which boosts their energy production. The idea is still being tested, but early results in cognitive recovery and mood stabilization are promising enough to keep labs busy. Then there's breathwork. Slowing your breathing activates the parasympathetic nervous system and lowers cortisol levels, creating a cellular environment that supports mitochondrial recovery. Deep breathing also improves oxygenation. Obvious, yes, but crucial for mitochondria, which need a steady oxygen supply to produce ATP. Think of it as filling the tank, slowly and intentionally. And if you're someone who experiences burnout, not just fog but the full-spectrum depletion of drive, joy, and stamina, mitochondria might be central to your story. Burnout isn't just mental. It's physical, biological, and often rooted in long-term energy imbalance. Your ATP reserves shrink. Your body tries to conserve energy. You lose the ability to bounce back after effort. Mitochondria are trying to keep up, but the demand never stops. Here's the hopeful bit: they are resilient. Even after extended dysfunction, mitochondria can recover. They can divide, renew, and rejoin the cellular dance. It takes time, rest, nourishment, patience, but they're not gone, they're just overwhelmed. So the next time your mind feels like it's running on low-resolution mode, when names escape you and your thoughts drift like fog on a quiet lake, take a breath. Move a little. Eat something real. Rest without guilt. You're not lazy. You're not broken. Your mitochondria are simply sending a message. And under the haze, they're still working, still trying to lift the fog from within. Your energy used to be effortless. Remember that, jumping out of bed as a kid, running just because, thinking clearly without caffeine, recovering from a sleepless night in a single afternoon nap. But somewhere along the way that shifted. Now energy takes effort. Your days begin with a slow climb and end in a quiet unraveling, and you've probably chalked it up to getting older. And you're right, but the reason might be more microscopic than you realize. The steady drain you feel is not just life's natural wear and tear. It's your mitochondria aging too. Mitochondrial decline is one of the central theories of aging. Not the only theory. Historians still argue whether DNA damage, telomere shortening, or metabolic drift deserves top billing. But mitochondrial dysfunction is consistently at the heart of it all. Because without energy, even the most finely tuned body systems falter. Aging isn't just about years passing, it's about power slipping, a slow, cellular dimming of the lights. Inside your cells, aging mitochondria begin to lose their structural integrity. Their inner membranes become less efficient, the once-tidy crystals start to flatten or swell, and the proteins responsible for making ATP lose their shape. It's like rust forming on gears, small at first, then pervasive. Energy output drops. ROS production goes up. The balance tips from function to dysfunction. You don't notice it all at once. It begins subtly: slower wound healing, sluggish recovery from illness, mental fatigue that lingers longer than it used to. But over time, the consequences mount. Tissues that rely heavily on energy, like muscles, brain, and heart, begin to show signs of wear. You may feel it as brain fog, aching joints, or that frustrating inability to recover like you used to. And here's a key point: aging mitochondria don't just underperform, they sometimes become toxic. They leak reactive oxygen species, they hoard calcium, and worst of all, they fail to die when they should. Your cells try to clean them up through mitophagy, but over time, that system slows down too. Old mitochondria linger like grumpy houseguests, draining resources and fouling the environment for the younger ones. This accumulation of dysfunctional mitochondria is called mitochondrial senescence, and it's a quiet thief. It doesn't cause dramatic damage all at once, it just makes everything a little worse, a little slower, a little less efficient. This slow decay is why researchers refer to mitochondria as both the drivers and victims of aging. They cause it, and they suffer from it too. The free radical theory of aging, once the dominant idea, suggested that ROS were the main culprits. That mitochondria, by producing these unstable molecules, effectively poison themselves and the cell around them. But now we know it's not quite that simple. ROS are part of normal signaling. It's not their presence but their imbalance that causes harm. In youth, cells neutralize ROS efficiently. In age, that balance slips. Antioxidant defenses falter. Damage accumulates. One of the most visible signs of mitochondrial aging is in your muscles. As mitochondria degrade, muscle fibers shrink and weaken, a process called sarcopenia. This isn't just about aesthetics. Loss of muscle mass is linked to frailty, falls, and decreased metabolic health. You're not just getting softer, you're losing cellular horsepower. Strength training and movement help, yes, but only if your mitochondria can still respond. And for that, they need maintenance. The heart, too, pays a price. Cardiac muscle is packed with mitochondria. As those mitochondria age, their rhythm falters. ATP production drops, calcium handling goes awry, and the risk of arrhythmias increases. In aging hearts, cells can even become energetically starved, leading to heart failure in extreme cases. Not from clogged arteries, but from cellular engines that just can't keep up anymore. And then there's the brain, where aging mitochondria contribute to cognitive decline. We talked about fog before, but now it deepens. Aging neurons lose mitochondrial support. Synaptic plasticity declines. Learning slows. Even memory formation suffers. Mitochondrial dysfunction has been implicated in Alzheimer's and Parkinson's diseases. More than just energy loss, it's miscommunication, misfolded proteins, and cellular confusion. But here's the strange part. Some aging mitochondria fight back. In certain cells, under specific conditions, mitochondria can upregulate their repair pathways. They can increase expression of protective enzymes. They can fuse with healthier mitochondria to dilute damage. Aging isn't always a straight line downward. There are bumps, loops, even moments of renewal. Researchers call this mitohormesis, the idea that mild stress can trigger a rebound, a strengthening, a rebalancing. Fringe experiments in mice have shown that caloric restriction and interval fasting can extend lifespan partly by preserving mitochondrial function. In one study, older mice on a reduced-calorie diet had mitochondria that looked and functioned more like those of younger animals. Their ATP output was better. Their ROS levels were lower, and their tissues showed less damage. Aging slowed, not reversed, but softened. There's also growing interest in compounds like NAD+ boosters and senolytics, drugs that target aged or dysfunctional mitochondria. The idea is to either recharge their energy pathways or clear them out entirely. It's still early days, but human trials are underway. And while no pill can stop the clock, these tools may one day help you age with energy instead of just withered time. For now, your best tools are the simple ones. Move often, sleep well, eat clean. Challenge your mitochondria with occasional stress, exercise, fasting, heat, cold, and just as importantly, rest. Because the process of mitochondrial repair, autophagy, biogenesis, antioxidant response happens most efficiently during recovery, not during hustle. And as you lie here now, curled quietly beneath your blanket, your cells are still working. Even your older mitochondria, the tired ones, haven't quit. They're still spinning, still folding ATP together like origami, still doing their best to keep your inner world alight. They're dimmer than they used to be, slower, but they're trying. And tomorrow, if you take a walk or skip that third cup of coffee or fall asleep 10 minutes earlier, they'll take that as a gift, a little breath of support, a reminder that even in aging there's still power, still fire, still time. There's a woman you've never met who lived long before your ancestors built anything resembling a house, long before written language, farming, or fire pits. She never knew she would become a scientific celebrity, or rather, a symbol. She left no name, no diary, no ruins. All she left behind was a line of mitochondria, handed down through the maternal chain across tens of thousands of generations. And because of that, she has a name anyway, Mitochondrial Eve. Now, before you picture her in biblical robes, know this: she wasn't the first woman or the only woman of her time. She wasn't some genetic Adam's partner, holding hands in a Garden of Eden. She was just one of many women living roughly 150,000 to 200,000 years ago, somewhere in East Africa. But her mitochondrial lineage is the only one that made it through to every human alive today. Why? Because all the other maternal lines eventually ended. Some had daughters who didn't reproduce. Others had mitochondrial mutations that stopped the line cold. Over time, Eve's line survived, not through greatness or strength, but through unbroken biological chance. Scientists piece this together by analyzing mitochondrial DNA from people all over the world. Since mitochondria are passed only from mothers to children, they act like a genetic time capsule. Unlike nuclear DNA, which gets shuffled and recombined every generation, mitochondrial DNA changes slowly and predictably. Mutations accumulate over time, creating distinct haplogroups, branches in the maternal family tree. And when you trace them all backward, following each fork through time, they converge at a single point: one woman, one lineage. Historians still argue whether this convergence reflects population bottlenecks, selection pressure, or random drift. Some say it paints an overly simplistic picture of human origins. After all, mitochondrial Eve wasn't alone, and she wasn't special. She was just lucky, in the most profound biological sense of the word. But the metaphor endures. She is, in a way, the mother of us all. Her mitochondria have powered human hearts through ice ages and deserts, wars and wanderings, births and deaths. They lit the minds of poets and hunters, fueled migrations across oceans, and now keep your phone charging, coffee-sipping self alive. Still humming, still folding ATP, still spinning those ancient gears. Your mitochondrial DNA, while nearly identical to hers, carries tiny mutations, markers of the journey. Scientists can tell whether your maternal line came through Europe, Asia, or the Americas based on these mutations. Each one is like a breadcrumb along a prehistoric trail, pointing back to one of the original haplogroups. You're not just a person, you're a path. And not all haplogroups are equal. Some appear in only certain regions. Others are more diverse. One group, L0, is thought to be the most ancient, still common among Khoisan people of southern Africa. Others, like H, U, and J, spread across Europe and the Middle East. These aren't just genetic curiosities. They hold clues to human migration, adaptation, and resilience. There's even fringe speculation that certain haplogroups might influence health. Some studies suggest people with certain mitochondrial backgrounds may be more prone to neurological diseases or better adapted to high-altitude living. Tibetan Highlanders, for instance, have mitochondrial variants that help them extract more energy from thin air. But the science is still young, and the genetics of health are never simple. In forensic science, mitochondrial DNA is gold. It can survive where nuclear DNA fails, burned tissue, degraded samples, ancient bones. It helped identify the remains of the Romanovs, the last imperial family of Russia. It's been used to trace the ancestry of famous mummies and solve modern cold cases. Because it's so durable, your mitochondrial DNA is more likely to outlive you than your name. And here's a strange little tidbit. In rare cases, mitochondrial DNA has been found to be paternally inherited, yes, from fathers. Only a handful of such cases exist, and scientists aren't even sure how it happens. Perhaps a sperm's mitochondria sneak in and escape destruction, or maybe it's a cellular accident. But these cases are so rare they're considered anomalies. The maternal rule holds firm, almost universally. Still, even mitochondrial Eve had a shelf life. She didn't live forever, and eventually, her own mitochondria aged and died with her. But her daughters carried copies and theirs, and so on, until one day, you. Lying here in a dark room, listening to a sleepy science story, quietly poolsing with the same mitochondria that kept her walking. If she knew, if mitochondrial Eve had any inkling that her cells would be powering the thoughts of a species that landed robots on Mars and invented bubble tea, she'd probably laugh, or blink in complete confusion. She was a woman of her time, just trying to stay alive, and in doing so, she gave you your fire. There's something comforting in that. The idea that through all the randomness of evolution, one tiny loop of DNA made its way through thousands of years just to end up inside your cells. It doesn't make life any easier, but it does make it more connected, more real, more shared. So as you drift tonight, tucked into your own corner of history, remember, there's a line from your mother to her mother, to 100 mothers more, all the way back to someone who never knew her name would echo forever in your cells. And her mitochondria, they're still glowing, faint and faithful inside you now. Somewhere deep in the early development of a human embryo, a quiet biological betrayal takes place. The father's mitochondria, carried in the tail of the sperm, packed with just enough energy to swim, are left outside. They never make it in. Or if they do sneak past the gate, the egg immediately tags them for destruction. No mercy, no second chances. Your mother's mitochondria are welcomed in with open arms. Your father's, immediately shredded. And it's not just cold-hearted cell protocol. It's deliberate. Mitochondrial DNA is maternally inherited, and the biological system ensures this by actively excluding or destroying the male contribution. But this raises a strange, slightly unfair truth. Men do not pass on their mitochondria, ever. No matter how many kids they have, their mitochondrial line dies with them. It's a kind of genetic dead end that, on the surface, seems trivial. But dig a little deeper and you find that it carries strange implications, especially when it comes to male fertility. While men don't pass on their mitochondria, they still rely on them intensely. Sperm cells are among the most mitochondria-dense cells in the body. Packed into the midpiece, these mitochondria give the sperm the energy to propel itself toward the egg. No fuel, no motion. No motion, no baby. But here's the kicker. If those mitochondria aren't working properly, if they're producing too many reactive oxygen species or not enough ATP, the sperm may still be formed, may still look perfectly normal under a microscope, but it won't move. Or it will move too slowly or in the wrong direction. Fertilization fails and no one quite knows why. Historians still argue whether this mitochondrial exclusion is simply a quirk of evolution or a deliberate defense mechanism. Some propose that it evolved to avoid mitochondrial warfare. If both parents contributed different mitochondrial DNA, the resulting cell might end up with incompatible organelles, fighting for dominance. Better to choose one. And since eggs are larger, more resource-rich, and loaded with quality control mechanisms, maternal mitochondria got the job. This maternal-only inheritance has consequences. Because mitochondrial DNA never gets mixed or recombined, it's clonally inherited. Mutations in a mother's mitochondrial DNA are passed on wholesale to all her children. If a mutation affects males but not females, say, by impairing sperm motility or testicular function, it still gets passed along, because the female carriers don't suffer and thus don't get filtered out by natural selection. This leads to a fascinating and a little tragic concept called the mother's curse. It's the idea that harmful mitochondrial mutations can accumulate in the male line, unchecked, because natural selection doesn't see their impact if females aren't affected. It's like a genetic blind spot. The mutation rides through generations invisibly, quietly undermining male fertility from the inside. Some studies in animals back this up. In fruit flies, certain mitochondrial mutations reduce male fertility without affecting females. In mammals, there's early evidence that links mitochondrial mutations to lower sperm count and reduced motility. In humans, the research is still unfolding, but clues are emerging. Men with certain mitochondrial haplogroups show differing sperm performance. Certain mutations are overrepresented in infertile populations. And some mitochondrial diseases are more severe in males, despite being inherited from mothers. Here's a quirky little twist. While mitochondria in sperm aren't passed to the embryo, they do influence fertilization outcomes. Some fertility clinics are now looking at mitochondrial function as part of male fertility assessment. Not just the number or shape of the sperm, but how well their mitochondria are producing energy. In the same way we test an engine's horsepower, we're now testing the spark within. And beyond fertility, mitochondrial dysfunction may play a role in other male-specific issues, like testosterone regulation, prostate health, and even certain mood disorders. Testosterone synthesis, for instance, is an energy-intensive process that occurs in mitochondria within Leydig cells. If those mitochondria are sluggish, hormone levels may drop, leading to fatigue, low libido, and other symptoms that get shrugged off as just aging. Of course, men aren't helpless in this story. While they can't pass on their mitochondria, they can optimize their own. Exercise, diet, sleep, stress, all the same rules apply. And some researchers are even exploring the idea of mitochondrial rescue in sperm, supplementing or restoring mitochondrial function to improve fertility outcomes. Early-stage studies are playing with antioxidant therapies, mitochondrial boosters, and even mitochondrial transfer. Yes, replacing faulty sperm mitochondria with donor ones. We're not quite at the sci-fi stage, but the groundwork is being laid. Interestingly, some species have different systems entirely. In certain shellfish and insects, paternal mitochondria do get passed down, or both parents contribute mitochondrial DNA. The rules we think of as universal in humans are not so absolute in the animal kingdom. Evolution likes to experiment. And what about future humans? Some fringe thinkers suggest we may one day reprogram mitochondrial inheritance, allowing fathers to pass on their mitochondria, or blending both parents' mtDNA. It's a controversial idea, not just biologically but ethically. Would combining mitochondria create compatibility issues? Would it disrupt the delicate balance cells rely on? No one really knows, but the fact that we're even asking the question shows how far mitochondrial science has come. So here you are, lying in the dark, your cells gently glowing with mitochondrial activity, all of it from your mother's side. If you're a man, your contribution ends with you. If you're a woman, you might one day pass that line forward. But regardless of sex, your mitochondria still power every breath, every blink, every thought. Even if they don't live on, they matter now. And that's enough. Not everything has to be passed on to be powerful. Let's take a detour away from humans and into the mossy corners of the forest floor, where you'll find one of our oldest, oddest relatives, fungi. Mushrooms, yeasts, molds, the quiet decomposers of the natural world. You might not think you have much in common with a shiitake or a patch of mildew, but when it comes to mitochondria, you're practically cousins. And that's not just poetic metaphor, it's cellular fact. Fungi, like animals, are eukaryotes, meaning their cells have nuclei and, yes, mitochondria. And while their lifestyles are wildly different from ours, their mitochondria follow surprisingly familiar patterns. They generate ATP. They run the electron transport chain. They divide and fuse. They even undergo mitophagy when damaged. Look at a fungal mitochondrion under a microscope, and you might not even recognize it as non-human. But here, where it gets weirder, fungal mitochondria are sometimes more flexible than ours. Certain fungi can survive in low oxygen or high-stress environments by shifting their mitochondrial output. Yeasts, like Saccharomyces cerevisiae, can switch between aerobic respiration and fermentation depending on what's available. They use oxygen when they can but fall back on fermentation when they must, kind of like switching from a Tesla to a bicycle in a blackout. This metabolic flexibility is what allows fungi to colonize almost any environment, from icy glaciers to the warm folds behind your bathroom tiles. And their mitochondria are key players in that adaptation. In some species, mitochondrial genomes are massive, even larger than those in plants. Armillaria, a genus of forest fungi sometimes called the honey mushroom, has mitochondrial DNA orders of magnitude larger than ours. And yet, it functions with eerie precision, helping these fungi live for thousands of years and cover entire forests underground. Historians still argue whether these expansive mitochondrial genomes offer any advantage. Are they just genomic clutter? Or do they hold untapped regulatory secrets? Some researchers suspect these large genomes may provide resilience in changing environments, allowing fungi to quickly adjust their internal machinery when conditions go sideways. And get this, some fungal mitochondria don't follow the rules of inheritance. In humans, as you know, mitochondrial DNA is passed maternally. But in many fungi, inheritance is biparental or even random, depending on the species. When two fungal cells mate, yes, fungi mate, albeit in extremely fungi-like ways, their mitochondria may mix, compete, or one type may be selectively degraded. In some cases, the mitochondria actually fight, with one strain's organelles outcompeting the others. It's a microbial game of thrones, and it all happens silently in rotting logs and damp soil. Here's a quirky tidbit. In baker's yeast, scientists have observed petite mutants, cells with defective mitochondrial DNA that can no longer respire. These mutants survive by fermenting sugar, but they're fragile and energetically inefficient. Researchers use them to study mitochondrial dysfunction and aging, because yeast replicates quickly and shows clear signs of mitochondrial stress. In other words, we're learning about human cellular burnout from bread. Fungi have even been used to test mitochondrial targeting drugs. Since their energy systems mimic ours in crucial ways, fungal cells make excellent models for early-stage pharmacology. If a new compound boosts ATP production or reduces ROS in yeast, it might do the same in human cells. And if it kills the fungus, well, that's good news too, particularly in an age of rising antifungal resistance. And while we're on strange crossovers, consider this. Fungi can get mitochondrial diseases too, just like us. Mutations in their mitochondrial DNA can make them slower, weaker, or unable to thrive in certain conditions. In agricultural settings, fungal mitochondrial dysfunction can impact crop yield, soil health, and even global food supply chains. Your toast depends on mitochondrial integrity more than you might think. There's also a philosophical undercurrent here, because the more we study fungal mitochondria, the blurrier the line becomes between them and us. We share an ancestor, after all. At some point over 1 billion years ago, a single-celled organism engulfed a bacterium, and both animals and fungi descended from that union. Our mitochondria and theirs evolved in parallel, diverging but never severing the common thread. Some fringe scientists have even proposed that fungi might hold clues to longevity. Certain long-lived fungal colonies, like the aforementioned Armillaria, show remarkable mitochondrial resilience. No swelling, no fragmentation, no ROS overload even after thousands of years. Could their organelles teach us something about how to delay aging? No one knows yet, but you can bet someone somewhere is sequencing a mushroom right now to find out. And let's not ignore the poetic angle. Fungi are nature's recyclers. They decompose the dead and feed the soil, all while humming with mitochondrial life. Their quiet work sustains entire ecosystems. And the same type of organelle that powers your heartbeat also powers the growth of a mushroom pushing up through forest loam. That's not just a biological coincidence. It's a kind of deep kinship. So tonight, as you lie there thinking you're separate from the damp, strange world outside your window, remember this. You share a molecular blueprint with the mushrooms. Your cells, like theirs, house ancient engines. Your energy comes from the same origin. And somewhere out there in the moss or the mulch, your cellular cousins are doing their quiet work, one ATP molecule at a time. You're not just human, you're part fungal, and that's not weird, it's wonderful. Let's take a step into the future now, into a lab where things are glowing that shouldn't be, where scientists in crisp coats peer into Petri dishes lit with soft blue light, and where mitochondria are no longer just being studied. They're being hacked. Because while your natural mitochondria have been refining their skills for 1 billion years, modern science has decided that's not quite fast enough. Welcome to the age of Frankenstein cells, synthetic mitochondria, engineered hybrids, and ambitious attempts to upgrade life from the inside out. At the core of it all is one idea. If mitochondria power the cell, then controlling mitochondria might let us control the cell's fate. And if we can improve or replace faulty mitochondria, we could cure diseases, slow aging, or even design more resilient bodies. Sounds a little like science fiction, but it's already happening. Take synthetic mitochondria. Yes, we're literally building them. In recent years, researchers have developed artificial mitochondrial membranes that can house working copies of key enzymes and pump protons, just like the real thing. The goal is to create organelle-like structures that can be inserted into cells and take over energy production when native mitochondria fail. A mitochondrial prosthetic, if you will. We're not at the level of plug and play just yet, but early prototypes show promise in lab settings. Tiny vesicles mimicking mitochondrial function have restored energy production in damaged cells. Think of it as a backup generator being wheeled in during a blackout. Not as efficient, perhaps, but enough to keep the lights on. There's also work being done on mitochondrial transplantation, a kind of cellular organ donation. Scientists extract healthy mitochondria from a donor cell and inject them into a damaged one. The hope is that the new mitochondria will integrate, restore function, and maybe even outcompete the bad ones. In animal models, transplanted mitochondria have helped repair heart tissue after injury and improved muscle function in degenerative diseases. Historians still argue whether transplanted mitochondria can truly fuse and cooperate long-term, or whether the benefits are temporary. But the fact that we can even get them into another cell and watch ATP production spike, that's groundbreaking. And then there are the hybrids, cybrids, short for cytoplasmic hybrids. These are cells created by merging the nucleus of one cell with the cytoplasm and mitochondria of another. Scientists use cybrids to study how different combinations of nuclear and mitochondrial DNA interact, how mismatched genomes affect energy production, disease expression, or cellular aging. Sometimes the results are harmonious. Other times it's a disaster. The wrong combo can lead to energy imbalances or trigger cell death. This highlights something profound. The relationship between your nuclear DNA and mitochondrial DNA is co-evolved. They're used to each other. When you introduce a stranger into that relationship, things can get awkward fast. Still, the idea of editing or replacing mitochondrial genomes is so tantalizing that researchers are pushing past the awkwardness. Enter mitochondrial gene editing, a field that's finally gaining traction. CRISPR, the tool that revolutionized genetic engineering, doesn't work well on mitochondria yet, but alternatives are emerging. One of the most promising is mitochondrion, a kind of molecular scissors designed to target and eliminate mutated mitochondrial DNA. By removing the bad copies, healthy ones can expand and restore function. It's like pruning a diseased branch so the rest of the tree can grow strong. Early experiments show this technique may help treat mitochondrial diseases that, until now, had no cure. Conditions like Lee syndrome or MELAS that steal energy from the body one cell at a time. And while we're talking about tinkering, let's dip into the slightly absurd but very real realm of biohacking. There are individuals out there, often not scientists, who are experimenting on themselves with supplements, extreme diets, cold plunges, and off-label drugs to boost their mitochondria. Some claim enhanced focus, improved energy, even delayed aging. Others end up with very expensive pee. The science behind most of these hacks is thin, but the enthusiasm is undeniable. One fringe experiment involved inserting bacterial enzymes into human cells to rewire how mitochondria process glucose. The goal? Supercharge energy production. The result? So far, only modest improvements. But the implications are massive. If we can make mitochondria more efficient, we might improve everything from athletic performance to cognitive endurance. It's mitochondrial performance enhancement, and someday it could be personalized. Of course, there are ethical landmines all over this field. What happens when we create cells with super mitochondria? Could enhanced people emerge with better energy systems, faster healing, greater endurance? If so, who gets access? Who decides what's natural? These aren't just hypothetical questions. They're already being asked in biotech boardrooms and bioethics forums. And what about synthetic life? Could we build a fully artificial cell, mitochondria included, from scratch? Some scientists think it's possible. The Minimal Cell Project is trying to define the smallest number of genes needed to support life. Once that's known, building from the ground up becomes much more feasible. A cell powered by a fully synthetic mitochondrion. That's the holy grail of cellular engineering. But let's take a breath. For all our ambition, we still don't fully understand the language of mitochondria, how they signal, how they sense damage, how they adapt to stress. We're learning, but the system is elegant and deeply complex. Each tweak risks unintended consequences. Still, the fact that we're even in this conversation is astonishing. From mistaking mitochondria for parasites in the 19th century to now attempting to rebuild them in labs, science has moved fast. Faster than mitochondria can replicate in some cases. And tonight, as you lie back with your natural, unedited mitochondria softly humming in each cell, think about what's coming. Not just pills or therapies, but a time when we might choose what kind of mitochondrial life we live. A time when cellular power can be tuned like a thermostat. When fatigue, disease, even aging might be optional, or at least negotiable. The future isn't made of flying cars. It's built on energy. And that energy still starts here, in the tiny bean-shaped engines inside you. Upgraded maybe, but still mitochondrial at heart. Now picture yourself floating even higher, above the planet, past the weather, beyond the stratosphere, out into the cold, black quiet of space. The Earth is just a glowing marble below you. There's no gravity tugging on your muscles, no wind on your skin. And inside your body, a very different kind of silence. Your cells are disoriented, your fluids are drifting, and your mitochondria, they're not happy. Turns out space messes with mitochondria. In ways we're only beginning to understand, the very engines that power your cells begin to glitch when you leave the planet. Energy production drops. Reactive oxygen species go up. Mitochondrial DNA mutates more frequently. And the longer you're out there, the worse it gets. NASA has been studying this for years, especially since the rise of long-term missions aboard the International Space Station. Astronauts often return to Earth with muscle atrophy, bone density loss, cognitive changes, and altered immune function, all of which have mitochondrial fingerprints. These aren't just the results of floating around. They're symptoms of a deep cellular recalibration in response to microgravity, radiation, and isolation. In 2019, NASA released findings from the twin study, a groundbreaking project comparing the physiological changes between astronaut Scott Kelly, who spent nearly a year in space, and his identical twin, Mark, who stayed on Earth. One of the most striking discoveries: altered mitochondrial function in Scott. His body shifted toward increased inflammation, reduced oxidative phosphorylation, and signs of cellular stress. His mitochondria had to adapt to a strange new environment, and they didn't do it quietly. Historians still argue whether microgravity itself causes mitochondrial dysfunction, or whether it's the cosmic radiation, the disrupted circadian rhythms, or the psychological stress of living in a metal 250 miles above Earth. Most likely, it's all of the above. But the result is clear. Mitochondria don't like space. In fact, some fringe researchers have started calling mitochondria gravity-sensitive organelles. That might be an overstatement, but it's not without merit. Under microgravity, mitochondrial shape and dynamics change. They fuse differently. They move differently. Their cristae look strange under electron microscopy. It's like they're confused, trying to perform familiar tasks in unfamiliar conditions. And then there's radiation. Earth's atmosphere protects us from most of the sun's high-energy particles, but in space, even short missions expose astronauts to increased levels of ionizing radiation. That radiation slices through cells, damaging DNA, including mitochondrial DNA, which has fewer repair mechanisms than nuclear DNA. The result: a higher chance of mutations, dysfunction, and long-term degradation. Mitochondrial damage from spaceflight isn't just a concern for astronauts. It's also a huge barrier to deep space travel. If we're ever going to send humans to Mars, they'll be in transit for months. And if their mitochondria start failing en route, even the best exercise regimens and protein bars won't help. To combat this, researchers are developing countermeasures: antioxidant-rich diets, pharmacological support, artificial gravity habitats, and even genetic screening to select candidates with more resilient mitochondrial profiles. Some of this sounds like sci-fi. Some of it is quietly already happening. Here's something a little more charming. Plants and microbes in space also show mitochondrial changes. Experiments with yeast aboard space stations reveal altered respiration rates and changes in mitochondrial morphology. Arabidopsis plants show shifts in mitochondrial gene expression when grown in microgravity. Even the simplest life forms, when unmoored from Earth's gravity, experience mitochondrial unease. And let's not forget the psychological side. Space messes with sleep. It messes with mood. And those changes ripple back into mitochondrial behavior. Just like on Earth, chronic stress in orbit means elevated cortisol, oxidative damage, and ATP inefficiency. Even the thrill of zero-G can't mask the fact that mitochondria are quietly freaking out. So what does all this mean for the rest of us with our feet firmly on the ground? Well, space turns out to be a kind of accelerated aging lab. The changes astronauts experience, mitochondrial decay, inflammation, muscle loss, mirror what happens to people over decades on Earth. Studying them helps us understand how to slow or reverse these processes back home. In a strange twist, the quest to keep astronauts functional may lead to better treatments for fatigue, frailty, and chronic illness here on Earth. One promising line of research is focused on mitochondrial-targeted antioxidants, like MitoQ or SS31, which are designed to enter mitochondria directly and mop up excess ROS. They're being tested both for space medicine and earthbound conditions like Alzheimer's, heart disease, and diabetes. If they work up there, they might work even better down here. There's also talk of hibernation-style metabolism for long space flights, a way to slow mitochondrial activity, reduce oxygen demand, and stretch energy reserves. Some animals do this naturally: bears, turtles, and even certain frogs. Their mitochondria downshift into low-output modes without shutting down completely. If we can mimic that, we might one day send people to Mars without burning through their biological batteries. Fringe idea alert. Some synthetic biologists are exploring whether we could engineer radiation-resistant mitochondria, drawing inspiration from extremophiles like Deinococcus radiodurans, a microbe that can survive nuclear blasts. Could we install its resilience into human cells? Maybe. Someday. Carefully. So here you are, far from zero gravity, grounded in the pull of Earth, breathing air that hasn't passed through a filtration system. Your mitochondria are at home. They know this rhythm. They understand gravity, light cycles, sleep cues. They perform better here. But it's worth imagining, if you ever did leave this planet, your mitochondria would come with you. They'd adapt as best they could. They struggle, yes, but they'd keep folding ATP as long as they could, in orbit, in transit, on another world. Because even in the silence of space, even when you're weightless and unmoored, the engines never stop. They flicker, they strain, they power every heartbeat against the stars. Imagine you're a detective, not with a trench coat and magnifying glass, but with pipettes, sequencers, and a centrifuge. Your crime scenes aren't alleys or rooftops. They're cells, tissues, fragments of bone centuries old. And the clues? Not fingerprints or shoe prints, but genetic whispers spiraling through the cell's tiny, tireless engines. Because when you want to find out what happened, who someone was, where they came from, or what disease they might carry, there's one source of evidence that almost always survives: mitochondrial DNA. It's durable, it's abundant, and it's often the last readable code when all else has decayed. That makes mitochondrial DNA one of the most powerful tools in forensic science, evolutionary biology, and modern medicine alike. But the twist is, these genetic breadcrumbs don't just point backward through ancestry. They also lead us into the tangled web of disease. Let's start with the disease hunters, the doctors and researchers trying to crack the code of conditions that defy easy explanation. When a patient presents with fatigue, muscle weakness, neurological symptoms, or mysterious metabolic problems, sometimes the nuclear genome shows nothing unusual. It's the mitochondrial DNA that holds the answers. That's because many rare disorders, especially those involving energy failure, trace back to mutations in the mitochondrial genome. There are over 300 known mutations in mtDNA that cause or contribute to disease. Some affect proteins in the electron transport chain. Others disrupt tRNA molecules, crippling protein synthesis. Because mitochondria are everywhere in the body, symptoms vary wildly depending on where the damage is worst. In some cases, it's the brain. In others, the muscles, or the heart, or the eyes. One mutation can cause vision loss in one person, seizures in another, and chronic fatigue in someone else. Diagnosis becomes a high-stakes guessing game, and mtDNA analysis becomes the only flashlight in the dark. Take Leber's hereditary optic neuropathy, a condition where sudden vision loss strikes in young adulthood. It's traced to a specific mitochondrial mutation that affects the optic nerve. Or MELAS syndrome, mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes, which sounds like a medical horror story because, well, it is. And then there's Kearns-Sayre syndrome, which causes progressive eye muscle paralysis, heart block, and other symptoms that might seem unrelated until you look at the mitochondria. Historians still argue whether certain mtDNA mutations are truly pathogenic or just unlucky variants, neutral until triggered by stress, aging, or another environmental factor. This uncertainty makes diagnosis a challenge. But as sequencing technology improves, doctors are beginning to identify mtDNA variants earlier, even in newborns or in people who show no symptoms yet. And it doesn't stop at rare diseases. Researchers are now digging into the mitochondrial contribution to common illnesses like diabetes, Alzheimer's, Parkinson, and even cancer. These aren't typically caused by mtDNA mutations alone, but mitochondria play supporting and sometimes starring roles. In Alzheimer's, for instance, dysfunctional mitochondria generate oxidative stress, fueling the buildup of amyloid plaques. In type 2 diabetes, mitochondria and muscle cells may lose the ability to process insulin signals properly, contributing to insulin resistance. Even cancer has a mitochondrial story. Some tumor cells shift their metabolism away from oxidative phosphorylation and toward glycolysis, a phenomenon known as the Warburg effect. But others maintain or even increase mitochondrial function. It varies by cancer type, and researchers are now using mitochondrial gene expression profiles to classify tumors and predict treatment response. That's right, your mitochondrial data might one day guide your personalized treatment plan. Some companies already offer mtDNA testing as part of genetic health panels. The information can point to potential energy disorders, guide lifestyle recommendations, or reveal inherited mutations you never knew you had. And then there's the forensic side. When all that's left of a body is a fragment of hair, tooth, or bone, nuclear DNA often fails, too degraded, too scarce. But mtDNA, there can be thousands of copies in a single cell. It's tougher, more numerous, and more likely to survive the ravages of time. That's how scientists identified the remains of King Richard III, unearthed from beneath a parking lot. That's how they sorted out the fate of Anastasia Romanov, untangled cold cases, and even challenged historical myths. Because mtDNA is inherited maternally, it can't prove individual identity on its own. But it can rule people in or out. It can place remains into a maternal lineage. It can help reconstruct missing persons. It's not the whole story, but it's a chapter many investigators can't live without. Here's a quirky side note. Ancient mtDNA has become a cornerstone of archaeology. From Neanderthals to Denisovans to early Homo sapiens, much of what we know about human prehistory comes from degraded mitochondrial samples recovered from caves, bones, or even ancient chewing gum. Yes, really. Researchers now use this data to map ancient migrations, trace interbreeding events, and understand how mitochondrial lineages rose and fell over millennia. And occasionally, those ancient sequences reveal surprises, like lineages that no longer exist today, or mysterious splits that suggest unknown human relatives, ghost lineages, whose mitochondrial trails vanish without a trace. These discoveries are rewriting our understanding of human evolution, all thanks to a tiny loop of DNA that refuses to die. Even now, new methods are being developed to extract and analyze mtDNA faster and more accurately. Portable sequencers, AI-driven pattern recognition, techniques that could one day allow a complete mitochondrial analysis from a single droplet of blood in under an hour, which means future doctors or detectives might diagnose energy disorders or solve crimes without ever leaving the room. But tonight, here you are, lying still in a quiet place, your cells softly buzzing, each one housing hundreds of mitochondrial genomes, your own archive, your own evidence, your own breadcrumb trail through time. They've been tracking your lineage, your health, your energy needs since the day you were born, silently, steadily, recording your story. And maybe, one day, someone might read it back. And now, we reach the edge of what is known and peer carefully over, not into science fiction exactly, but into the accelerating, uncharted now. Because mitochondria, once considered humble and unchangeable engines of the cell, are rapidly becoming the center of some of the boldest questions in biology. Not just how do we fix them, but can we farm them, clone them, replace them altogether? What does the future look like if we can fully control the powerhouse within? Already, research is rushing ahead. Scientists are exploring mitochondrial gene therapy, not just removing faulty mitochondrial DNA, but actively editing it to correct disease-causing mutations. Unlike nuclear DNA, which has been widely edited using CRISPR and other tools, mitochondrial DNA resists many of the usual techniques. It's locked away in a separate compartment, stubborn, circular, and protected by multiple membranes. But now, specialized editing tools like MitoZFs and DDD-A-derived cytosine base editors are being developed that can make precise changes, base by base. Imagine correcting a mitochondrial mutation in a single embryo before symptoms ever begin. That's not just a treatment, that's prevention at the blueprint level, and it's not hypothetical anymore. In a few rare cases, it's already been done. In parallel, biotech firms are racing to produce mitochondrial replacement therapies at scale. As we've touched on before, this involves swapping out the faulty mitochondria in a woman's egg with healthy ones from a donor, effectively creating embryos with 3 genetic parents, 2 for the nucleus, 1 for the mitochondria. It's already approved in some countries for specific cases of mitochondrial disease. But as the techniques improve, the conversation is shifting from "should we do this?" to "how often will we do this and for whom?" Historians still argue whether mitochondrial manipulation is a slippery slope toward eugenics or just another tool in the toolbox of modern medicine. Some worry about equity, who gets access, who decides what's normal. Others argue that if we can prevent suffering, we're ethically obligated to try. And looming above it all is the question of unintended consequences. Mitochondria have co-evolved with nuclear DNA for eons. Swap them out too freely, and you might introduce mismatches, tiny incompatibilities with outsized effects. That's why researchers are also looking into cytoplasmic compatibility, trying to identify mitochondrial nuclear pairs that work well together. It's like a genetic dating service, pairing the right organelles to avoid cellular tension. Because even if you swap in healthy mitochondria, they might not behave properly if they're in a nuclear environment they don't recognize. There's also talk of mitochondrial biobanks, banks of harvested, sequenced, quality-screened mitochondria from diverse donors. In the future, if someone needs replacement mitochondria, their doctor might order a compatible batch, matched by genetic markers the same way we now match for organ transplants or blood type. A kind of internal spare part catalog. Meanwhile, the private sector is looking at mitochondria through another lens entirely: performance enhancement. A new wave of startups is developing supplements, gene therapies, and injectable compounds that claim to supercharge mitochondrial output, boost endurance, sharpen mental clarity, delay aging. Some are snake oil, some are promising, and a few are already being tested in elite athletes or early clinical trials. One such compound, alamipretide, is being explored for its ability to stabilize the inner mitochondrial membrane, preserving crystal structure and reducing oxidative damage. It's shown benefits in aging mice and in early human trials for heart disease and mitochondrial myopathy. If successful, it could become the first widely used mitochondrial medicine. There are also whispers about synthetic organelles, not replacements but enhancers. Tiny compartments inserted into cells to support, augment, or even supervise mitochondrial activity. Think of them as mitochondrial wingmen. They're not meant to take over but to help out, handling overflow, stabilizing energy output during stress, buffering against collapse. Still early days, but the vision is there. And then there's the wild card: AI and synthetic biology. As computational models of mitochondrial behavior become more sophisticated, researchers can simulate how different genetic changes will affect ATP production, ROS generation, and cellular health. In some labs, AI is already designing novel mitochondrial proteins that don't exist in nature, proteins that might one day be printed and delivered into cells like microscopic upgrades. Of course, none of this is simple. Mitochondria are deeply integrated into the web of life. They respond to diet, stress, sleep, temperature, movement, light. They're not isolated machines, they're collaborators in the cell's symphony. Any attempt to control them must be subtle, respectful, and incredibly well-informed. And here's the strangest part. The more we try to manipulate mitochondria, the more they remind us of their autonomy. They divide independently. They have their own genome. They even initiate cell death when things go wrong. They are ancient tenets of our biology, and while we may someday improve them, we will never fully own them. But that's okay, because the goal isn't domination, it's harmony. It's giving these ancient powerhouses what they need to do their job better, for longer, with fewer stumbles and fewer scars. The more we learn, the more that looks possible. So as you close your eyes tonight, you lie at the threshold of that future. Inside you, right now, mitochondria are still spinning ATP from glucose and oxygen, like they've done since the first fish squirmed through ancient oceans. But out there, in glowing labs and humming sequencers, the next chapter is being written. A chapter where disease fades, where energy returns, where age slows. Where the powerhouse within isn't just explained, but reimagined. Section 21. The hum fades now, not because the mitochondria have stopped, but because you're starting to tune in more gently. We've been deep in the world of future tech, engineered cells and synthetic dreams, but now we drift back into your body, into your breathing, into the quiet rhythm of real life. Let's pause, not on the cutting edge, but at the core of what mitochondria mean for you here and now. Because while the science dazzles, it always comes back to something simpler: how your choices, daily, mundane human choices, shape the life and function of these ancient, stubborn engines. Every breath you take brings in oxygen not for your lungs but for your mitochondria. That's the whole point of breathing. Not so you can smell the ocean or sigh dramatically, though both have their place, but so that the oxygen can slip into your blood, hitch a ride to your cells, and land in the mitochondria where it waits patiently at the end of the electron transport chain. It's the final acceptor, the soft, invisible endpoint of the entire process. Without it, energy production halts in minutes. You don't just get sleepy, you collapse. That's why oxygen flow is so critical to mitochondrial health. It's not just about how much you breathe, but how well your blood delivers. Movement helps, so does posture. And yes, deep, regular breaths matter more than you think. This is why meditation and slow breathing can actually change your cellular chemistry. They lower stress, reduce oxidative load, and increase oxygen efficiency. Small habit, big shift. And then there's food. Not just the trendy kinds, although sure, leafy greens and wild-caught salmon will do you favors, but also how you eat when you eat and what you don't eat. Mitochondria run best on a balanced fuel mix: glucose, fats, and occasionally ketones. But when overwhelmed, say, by ultra-processed foods, constant grazing, or late-night binges, they get overloaded. The electron transport chain stalls, ROS rises, damage begins. It's not about perfection. You don't need to worship spirulina or count every almond. But understanding that every bite you take filters down to a mitochondrion is enough to nudge things gently in the right direction. Whole foods over processed, plants over packages, real over refined. Sleep too is non-negotiable. During deep sleep, your body engages in mitochondrial maintenance. Old ones are recycled, new ones are born, damaged proteins are removed. ATP production is recalibrated for the next day. Miss that window too often and the system backs up like a sink full of dirty dishes. Your energy crashes, your mood sways, your cells whisper, "Please, just rest." And what about light? That simple, everyday environmental factor you barely notice. Sunlight, particularly in the morning, sets your circadian rhythm, which directly affects mitochondrial efficiency. Cells that know what time it is perform better. They make energy when it's needed. They slow down at night. Artificial blue light at midnight. It confuses the system. Like running your dishwasher at 3 a.m. every night and wondering why the pipes groan. You don't need to become a monk or a biohacker. You just need to respect your mitochondria the way you'd respect a good barista. Don't demand too much, too fast, without offering something in return. Feed them. Rest them. Don't drown them in sugar. Let them breathe. They'll thank you by doing what they've done for 1 billion years, powering life with quiet consistency. And when you do challenge them, when you exercise, when you fast, when you dip into a cold lake or sweat in a sauna, they respond with adaptation. Hormesis, remember. The idea that stress, when small and well-timed, leads to strength. Mitochondria want a little challenge. They don't thrive in stagnation, but they also need recovery. It's not about grinding harder, it's about knowing when to sprint and when to sleep. There's poetry in it, really. That your energy, your resilience, your ability to think and feel and move comes from something so ancient, so microscopic, and yet so responsive to your choices. Mitochondria may be built from bacterial ancestors, but they're shaped by human behavior, your behavior. And here's something both beautiful and sobering. Your mitochondrial function affects the next generation. The better your mitochondrial health, the better your eggs, your sperm, your future child's energy blueprint. This is why maternal health matters so much, not just during pregnancy but before it. The mitochondria you pass on aren't just tiny, they're foundational. Scientists are still mapping exactly how your lifestyle today echoes forward into mitochondrial resilience decades from now. But already, the message is clear. The power within you is real, and it listens to what you do. So lie back, take one slow breath, and marvel at what's happening beneath your skin. Right now, in the quiet, your mitochondria are spinning, fusing, dividing, producing. They are, in many ways, the most ancient part of you, and the most alive. They don't need applause, they don't even need awareness. But a little kindness, a little rhythm, a little pause can go a long way. Because you don't have to biohack your way to better energy. Sometimes, all you have to do is cooperate. Now that you're cozy and perhaps a little floaty, let's wander back through time again. Not to the ancient ocean or early human lineages, but to the point where mitochondria weren't even mitochondria yet. Back when they were still free-living bacteria, drifting through primordial muck, just trying not to get eaten. Because even though we've talked about mitochondria like long-settled citizens of the cell, they weren't always part of the team. In fact, there was a time when they were invaders, or maybe guests who overstayed their welcome and wound up on the lease. Scientists believe this merger happened around 1.5 to 2 billion years ago, give or take a few hundred million. A larger, likely amoeba-like cell engulfed a smaller aerobic bacterium, probably some cousin of today's alpha protobacteria. But instead of digesting it, the host decided to keep it around, maybe because it was handy with oxygen, or maybe because it brought something to the table that was just too good to toss out. And thus, the endosymbiotic theory was born. We touched on it early in our journey, but now you can appreciate it in its full weirdness. A merger between two life forms, one swallowed by the other, leading not to destruction, but to cooperation. It's like adopting a stray cat who ends up managing your home Wi-Fi network. At some point, the boundaries blur. You stop asking who's in charge. This symbiosis didn't just work, it exploded. The host cell got access to efficient energy production. The bacterium got a protected environment and a steady food supply. Over time, genes from the engulfed bacterium migrated into the host's nucleus. The bacterium lost autonomy. It shrank, simplified, specialized. And somewhere along that long arc, it stopped being other and started being mitochondrion. Historians still argue whether the host cell was already complex when the merger happened, or whether the merger itself was the catalyst for complexity. In other words, did mitochondria make us multicellular, or were they just along for the ride? The consensus is leaning toward the first, that the energy boost from mitochondria allowed cells to grow larger, support more genes, develop specialized structures, and eventually evolve into the stunning zoo of forms we see today, from plankton to peacocks to people. And here's a strange fringe theory to lull you deeper. Some researchers have proposed that mitochondria might still be, in a very faint sense, semi-autonomous. That they're not just organelles, but something more, quasi-life forms with their own agendas, their own rhythms, maybe even rudimentary forms of decision-making. It's not about consciousness, of course, but about agency. The idea that mitochondria respond dynamically to their environment, making what could be called choices, based on signaling and feedback loops. Take mitochondrial dynamics, for example. They're not just passively existing, they fuse and divide with purpose. Fusion can help dilute damage, allowing weaker mitochondria to combine with healthier ones and share resources. Fission can isolate the damaged bits, flagging them for disposal. It's not random, it's responsive. And some scientists think this behavior mimics the early forms of intelligence, not thought, but adaptive behavior at a microscopic scale. It's this blend of ancient bacterial independence and deep cellular integration that makes mitochondria so hard to categorize. They're not like the other organelles, born from the host. They're aliens who integrated so well we stopped noticing. And maybe that's what makes them so vital. Their origins make them sensitive. Their independence makes them responsive. They are the perfect interface between the outside world and the inner machinery of life. They sense oxygen levels, nutrient availability, hormone changes, cellular stress. They're the first to know when something's off, and often the last to give up when things go bad. They're also incredibly conserved across species. The mitochondria in your cells aren't wildly different from those in a cow, a whale, or a house cat. That's why so much mitochondrial research is done in model organisms: mice, flies, even worms like C. elegans. These creatures give us real insight into how human mitochondria behave under stress, during aging, or in disease. One particularly compelling experiment involved tweaking the expression of certain mitochondrial genes in C. elegans. When researchers reduced activity in a specific complex of the electron transport chain, the worms actually lived longer. It seemed paradoxical: less energy, more life. But the theory is that mild mitochondrial stress triggered protective pathways, ramping up antioxidant defenses, improving protein folding, and generally making the cells more resilient. Mitohormesis in action. Similar results have been seen in other animals. Slight impairment of mitochondrial function, just enough to challenge the system without breaking it, can increase lifespan, boost resistance to stress, and even improve memory in older animals. Again, it's not about perfection. It's about pushing the system gently, keeping it alert, never letting it stagnate. And here's the kicker: the same principles apply to you. Mitochondria don't want to be pampered. They want to be used. They want challenge, recovery, rhythm. They don't want endless stimulation or chronic stress, but they also don't thrive in stillness. They were born in conflict and forged in partnership. They expect a little friction. That's why practices like fasting, exercise, and cold exposure seem to work at a cellular level. They recreate the environment mitochondria evolved in, a world of cycles, of feast and famine, effort and rest, light and dark. A world that expected cells to adapt, not just survive. So tonight, as your cells settle into their night rhythms, as ATP production shifts into maintenance mode and old mitochondria are marked for recycling, remember where they came from. Not a factory, not a blueprint, but a bacterium who struck a deal. And in keeping their end of that deal, your mitochondria became the flame that still burns at the heart of every living thing. The fire in your cells might be ancient, but that doesn't mean it burns evenly in everyone. Some people run hot, brimming with energy, glowing with vitality, bouncing up hills without coffee. Others move through life like they're dragging invisible weights, struggling to think clearly, stay awake, or recover from even small exertions. And in many of these cases, behind the scenes and beneath the bloodwork, there are signs pointing to one subtle culprit: mitochondrial dysfunction. Not disease, exactly. Just a creeping, uneven burnout of the body's internal battery pack. You've likely heard the term mitochondrial disease before, and maybe imagined it as something rare and genetic, which it can be. But the real twist is that mild to moderate mitochondrial dysfunction might affect millions of people, showing up not as a full-blown diagnosis, but as chronic fatigue, brain fog, exercise intolerance, and even vague symptoms like dizziness, low mood, or unexplained muscle pain. For some, the symptoms are crushing. Think of chronic fatigue syndrome, now more often called myalgic encephalomyelitis, or MECFS, a condition marked by profound exhaustion, unrefreshing sleep, and physical crashes after even minor activity. It's frustratingly hard to pin down, and for years it was dismissed as psychosomatic. But now, emerging research is pointing to mitochondrial bioenergetic failure in people with MECFS. Their cells simply can't keep up with energy demand. Mitochondria stall, recovery slows, and the whole system sputters. Historians still argue whether mitochondrial dysfunction is the cause of ME/CFS or just a byproduct of some deeper immune or neurological imbalance. But the mitochondrial fingerprint is consistent: reduced oxygen consumption, abnormal lactate levels, and a sluggish response to physical stress. These aren't people who are just tired. Their cells are literally underpowered. And it's not just ME/CFS. Mitochondrial dysfunction is being investigated in long COVID. 2. Another condition filled with brain fog, fatigue, and weird symptoms that don't follow clean diagnostic lines. Some researchers believe the lingering virus, or the body's response to it, might damage mitochondrial function, leaving survivors with cells that behave like they've aged decades in a matter of months. Even depression, anxiety, and neuropsychiatric disorders have mitochondrial threads. The brain is a voracious consumer of ATP, and when mitochondrial function dips, neurotransmitter production and synaptic firing can falter. That mental crash after a stressful day, that could be more than just burnout. It might be bioenergetic depletion, where your brain is literally running low on charge. In one study, people with major depressive disorder showed reduced mitochondrial activity in their neurons. In another, anxiety was linked with abnormal mitochondrial dynamics, more fragmentation, less fusion. These patterns aren't definitive yet, but they're shaping a new view of mental health. Not just chemical imbalances, but energy imbalances. And here, where things get personal. Maybe you've had those days where your body feels heavier, your thoughts foggier, your motivation harder to summon. You tell yourself it's stress or diet or the weather, and maybe it is. But beneath all of it, there's a good chance your mitochondria are playing a role, reacting to inflammation, nutrient levels, sleep quality, and even emotional stress. Because mitochondria don't just respond to biology, they respond to your experience. There's a phrase for this in research circles: mitochondrial allostatic load. It's the cumulative wear and tear on mitochondria caused by chronic stress. Your nervous system sends out a stress signal: fight or flight, worry, pressure, overstimulation, and your mitochondria respond. At first, they increase energy production to help you cope. But over time, with repeated stress and no rest, they start to degrade. Efficiency drops, damage accumulates. And the stress that once required energy becomes the thing that prevents you from making it. This is where lifestyle medicine starts to look a lot like mitochondrial therapy. Practices like mindfulness, breathwork, nature exposure, and good sleep hygiene aren't just wellness fluff. They're direct support for your mitochondria. They lower ROS production. They reduce inflammation. They restore metabolic rhythm. You may not feel it immediately, but on a cellular level, your mitochondria are breathing a sigh of relief. Some researchers are going even further, testing targeted supplements like CoQ10, alpha-lipoic acid, carnitine, and mitochondrial peptides like SS31 in clinical trials. The idea is to support the electron transport chain, mop up free radicals, and encourage biogenesis, the creation of new, healthy mitochondria. Results are promising but not miraculous. Think of them as gentle nudges, not magic bullets. But even without a pill, you have tools. Moderate movement, especially aerobic activity, stimulates PGC1 alpha, a master regulator of mitochondrial growth. Gentle intermittent fasting encourages mitophagy, cleaning out the sluggish organelles and making space for new ones. Sleep allows for repair. And connection, real, in-person human connection, lowers stress signals, letting mitochondria drop out of survival mode and return to steady, sustainable function. Of course, there are limits. Not all mitochondrial dysfunction can be reversed. Genetics still matter. Environment still matters. But the difference between stuck and stable often lies in those small, consistent choices, the ones that tell your cells, "Hey, I've got your back." So here you are lying still, perhaps still awake, perhaps hovering on the edge of sleep. Your body may feel tired. Your thoughts might still be racing. But deep inside, your mitochondria are doing what they can, folding proteins, moving ions, making ATP. Even if they're not perfect, even if they're struggling a bit, they're trying. And sometimes the most powerful thing you can do is simply give them space to recover. No fixing, no hacking, just soft, dark, still air and a little time. Section 24. The power within your cells isn't static. It shifts, flows, adapts. And nowhere is that adaptability more dramatic or more mysterious than in the process of development. From the moment you began as a single fertilized cell, your mitochondria were there, waiting in the wings. But they didn't take center stage right away. No, early development begins in whispers. The embryo is powered mostly by maternal stores, proteins, RNAs, a kind of biochemical trust fund passed down with the egg. The mitochondria in those early cells are quiet, dormant, holding their breath. Then something magical happens. As the embryo grows and cells begin to specialize, the mitochondria wake up. They start dividing, shifting shape, turning up the dial on energy production, not all at once and not equally. Muscle cells get one set of mitochondrial priorities, neurons get another. Even different regions of the brain end up with slightly different mitochondrial profiles. It's like each tissue negotiates its own power contract. How much, how fast, what fuel source to use. This dynamic process is called mitochondrial maturation, and it's guided by a complex dance of gene signals, nutrient availability, and cellular demand. Early on, cells rely heavily on glycolysis, breaking down glucose without much help from oxygen. But as mitochondria rev up, oxidative phosphorylation becomes dominant. It's more efficient, more sustainable, and crucial for complex tissues like the brain and heart. Historians still argue whether the timing of this mitochondrial shift affects long-term health. Some studies suggest that if mitochondria mature too early or too late, it can set the stage for metabolic problems down the line. The early environment, including oxygen levels, maternal nutrition, and even stress, might tweak how mitochondria behave forever. It's a kind of cellular imprinting, a long echo from the womb. This raises fascinating and slightly unsettling questions. What happens to the mitochondria in embryos exposed to high stress, to undernutrition, to pollutants or toxins? We're beginning to learn that early mitochondrial programming might influence not just energy levels but disease risk, diabetes, heart disease, even mood disorders. Your adult health might reflect not just your genes, but how your mitochondria were taught to behave before you were born. And this concept continues into childhood and adolescence. As kids grow, their mitochondria keep adapting. Puberty, in particular, is a time of massive mitochondrial upheaval. Hormones like estrogen and testosterone influence mitochondrial biogenesis and fuel preferences. In fact, sex hormones are mitochondrial regulators, which helps explain why certain mitochondrial diseases present differently in males and females. In boys, testosterone ramps up mitochondrial demand in muscle and brain tissues. In girls, estrogen appears to protect mitochondria, enhancing antioxidant defenses and supporting mitochondrial DNA repair. Some scientists believe this is one reason women tend to live longer. Their mitochondria age more gracefully. But estrogen levels drop after menopause, and with them, mitochondrial protection fades. This is one reason why postmenopausal aging accelerates. Bone loss, cognitive changes, cardiovascular risk, all of it may be tied, at least in part, to mitochondrial decline. Meanwhile, in men, mitochondrial stress accumulates differently. Because they don't pass on their mitochondria, There's less evolutionary pressure to maintain mitochondrial integrity in the male line beyond reproductive age. It's a subtle biological bias. Some researchers even argue this contributes to the shorter average lifespan in men. Still, there's a bright side. Throughout life, mitochondria remain trainable. Whether you're 5 or 95, your mitochondria can respond to changes in demand. They multiply when needed. They shift fuel sources. They clean up damage. They're not fixed assets, they're living systems. Which means that no matter how far along you are, change is always possible. Here's something quirky. In recent years, scientists have found that mitochondria even communicate with the nucleus more actively than we thought. They don't just take orders, they send status updates. These retrograde signals help the cell adjust to stress, infection, or injury. If mitochondria are struggling, they don't suffer in silence. They flag the problem. They try to help the cell adapt. In fact, this mitochondrial nuclear crosstalk is emerging as a key player in many conditions, from neurodegeneration to cancer to inflammatory disease. When the communication breaks down, the cell can't make good decisions. It keeps pushing forward with faulty instructions. But when the signals are clear, repair pathways kick in. Inflammation quiets. Harmony returns. You might not feel that harmony in daily life, but it's there. When you recover quickly from a cold, when your muscles bounce back after a hike, when your mood lifts after sleep, it's not luck. It's your mitochondria collaborating with the rest of you. And just as they rise to meet challenges, they also decline predictably with age. Mitochondrial DNA accumulates mutations. Membranes become less fluid. ATP production drops. This isn't failure. It's wear and tear. Like shoes losing tread or speakers losing clarity. But again, the decline isn't fixed. It varies person to person, lifestyle to lifestyle. In some centenarians, those lucky few who live past 100, mitochondria seem unusually resilient. Less fragmentation, lower ROS, more robust fusion. Scientists are studying their cells for clues. Is it genes, environment, just good luck? Probably all three. But it offers a hopeful message. Some mitochondria go the distance. And while you may not be planning to break longevity records, you are planning to wake up tomorrow with enough energy to move, think, and be. That goal alone is worth supporting. And the mitochondria you woke up with today are the same ones you'll fall asleep with tonight. Still adapting, still repairing, still humming quietly in the dark. So let them rest. Let them reset. They've powered every heartbeat you've had since the womb, and they're already preparing for tomorrow. By now, you've journeyed deep into the heart of your cells, down to the level where chemistry becomes identity, where movement becomes metabolism, and where something as small as a mitochondrial membrane can mean the difference between vitality and collapse. But as we draw closer to the edge of this long, sleepy trail, there's one more system to explore. A quiet, relentless cycle that touches everything. The death and renewal of mitochondria themselves. Because strange as it sounds, mitochondria are not immortal. They wear down, they falter, they fracture. And when they do, your body doesn't panic, it cleans them up. It rebuilds. This process is called mitophagy, a cousin to autophagy, and it's one of the most elegant forms of self-maintenance in your biology. Imagine a mitochondrion that's starting to misfire. Its membranes are leaky, its DNA is frayed, and it's spitting out reactive oxygen species like sparks from a damaged wire. Left unchecked, it would spread chaos, not energy. So the cell flags it. A molecular tag is placed like a condemned sign, and then slowly, gracefully, the damaged mitochondrion is engulfed and dismantled. Its components are either recycled or discarded. And in its place, a new one will eventually rise. This system works beautifully until it doesn't. With age, stress, and certain diseases, mitophagy slows down. Broken mitochondria accumulate. energy production drops. Oxidative stress increases. It's like leaving faulty batteries in a device. Things dim, flicker, malfunction. That's why boosting mitophagy has become a hot target in anti-aging science. Certain interventions seem to nudge the system. Intermittent fasting, for example, triggers a mild stress response that encourages cells to clean house. Exercise has similar effects. Even sleep appears to support mitochondrial recycling, especially in the brain. And more extreme methods, cold exposure, high-heat saunas, hypoxia training, also flirt with mitophagy stimulation. Not as punishment, but as signal: adapt or fall behind. Historians still argue whether ramping up mitophagy prolongs life or just improves the quality of it. Longevity studies in animals show promise, but humans are trickier. We live too long, do too many unpredictable things, and refuse to be placed neatly in lab cages. Well, most of us. But even if mitophagy doesn't buy us more years, it may offer us better ones, years with more energy, more clarity, more strength. And while the cleaning crew of mitophagy is vital, so is the creation crew. New mitochondria are born through biogenesis, a process triggered by signals like AMPK and PGC1 alpha, molecules that essentially tell your cells, we need more power. These are activated by, you guessed it, movement, fasting, and stress in gentle, controlled doses. When your body senses demand, it responds not by conserving, but by expanding its mitochondrial capacity. This is why athletes have more mitochondria per cell in their muscles than sedentary individuals. Why long-term meditators may have higher mitochondrial density in certain brain regions. It's not just about fuel, it's about building infrastructure to support that fuel use. And the best part, it's reversible. Fall into stillness too long and the system winds down. But move again, breathe deep again, and the sparks reignite. There's another layer to this, and it's strangely poetic. When a cell becomes too damaged, when its mitochondria signal that things are beyond repair, it doesn't just limp along. It commits to apoptosis, programmed cell death. And who triggers this self-sacrifice? The mitochondria themselves. That's right, these ancient powerhouses born from a bacterial invader now act as guardians of the cell's integrity. When they detect that damage has gone too far, they release proteins like cytochrome C, initiating a cascade that ends in orderly cellular shutdown. No inflammation, no mess, just a graceful exit. This isn't morbid, it's protective. Apoptosis prevents cancer. It clears out infected cells. It prunes unnecessary connections in developing brains. It's not the end. It's part of renewal. And again, it's your mitochondria calling the shots. You might wonder how a cell balances this dual role, creating energy for life, and deciding when that life should end. It's a tightrope, and it's one of the reasons mitochondrial dysfunction is so dangerous. When these decision points are corrupted, when mitochondria fail to initiate apoptosis properly, you can end up with rogue cells, cancerous cells, cells that ignore their expiration dates. On the other hand, too much apoptosis can lead to degeneration, cells dying off prematurely in the brain, heart, or muscles. Like most things in biology, it's not about extremes. It's about balance, a system that knows when to repair, when to build, and when to bow out. Some researchers are even exploring mitochondrial transplantation not just as repair, but as a way to reset cellular fate. Could inserting healthy mitochondria into damaged cells reduce their need to self-destruct? Could it extend their lifespan and restore their function? The early data is tantalizing. We're not just talking about slowing death, but reshaping it cell by cell. And as you lie still, with your own mitochondria quietly monitoring their environment, recycling what they must, creating what they can, and holding the line between function and failure, it's worth pausing to feel a little gratitude. Not the loud, celebratory kind, The quiet kind. The kind that recognizes effort without demanding perfection. Because they're not perfect. Not all of them. Some are tired, some are old, some are waiting for a signal that may never come, but they're still doing the work, every second, every breath. And if that's not a metaphor for being human, what is?